Hello, 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 hello. Welcome, everybody. Welcome to the stream. Hello, hi. Oh gosh, there's a good number of you out there. Welcome to the stream. Today I have a very special guest that will enable me to talk about something that I have been very afraid to tackle alone, so I'm very grateful for her being here. Please, special guest, introduce yourself.
Heyo! I'm Resta Makurabe. So, usually I introduce myself as pillow personified, cuteness bonafide, but today I'll be your friendly neighborhood bioinformatician.
Yeah. So, before I get into the bioinformatics stream proper, I just wanted to make an announcement to everybody in my audience. Hello to those of you who have come over from Resta's streams. I recognize a couple of names out there. Hello. I am actually going to be celebrating my 1-year content anniversary next week. I've got a bunch of fun things planned. It's a very, very special stream that I've put in quite a bit of work putting together, and I'm very excited to get to celebrate this amazing year of content creation here on YouTube. But today we have some more serious business to attend to, and that is to walk through the world of bioinformatics in a way that is, again, sort of a little different than you might get in a sort of lecture environment. And so, as always, my philosophy for this series of MolBio 101 streams is to bring you from a 101 naive area and then end in a really cool place where you're actually enabled to do some real world problem solving. And I actually wasn't going to do this on my own because I took bioinformatics when I was an undergraduate and I dropped out. Yes, I was so bad at this class that I had to drop it. I am forever a very mediocre Python programmer, and I was unfit, I think, to make it in this world of programming. So when I found that Resta has experience in the world of bioinformatics, I jumped on the opportunity to collab pretty much instantly. I think you had hopped into one video. We're able to answer all of my questions very clearly and cleanly, and I was like, this person biology's, and then we got to talking a little more, and I was like, oh my god, she's biology plus computers. That's exactly my weakness. And we got to planning this collab not too far after that encounter. I did get filtered out, that is true. 1 year is pretty good for a cell that usually only lives about 5 days. The sort of lore behind Phi the neutrophil is I do die every 8 days, and so every 8 days is a new clonal copy of Phi. Hello to all of you guys, there's so many of you, this is so exciting. Okay, so with that in mind, oh, I forgot. Resta, what is your exact experience in the field of bioinformatics, if you don't mind sharing a little bit before we get into the content proper?
So, uh, a bit before I got into bioinformatics specifically, I did some research in the field of environmental microbiology, especially work with marine microbes. And because it's very hard to culture them and characterize them, we often use bioinformatics, and that's kind of how I got my foray into it. Um, and it turns out I really liked programming, building tools for myself, and that whole engineering aspect of it.
Yeah, I think we actually
And
started talking around when I had released my Archaea video, if I remember correctly. And for me, that was my actual first foray into thinking about unculturable microbes, Archaea, all these really tough not pathogens, exactly the opposite of pathogens, these really tough, hardy microbes that are so mysterious to study. And when I heard that that's actually what you were like interested in, or that's where you at least did some work in, I was like, dang, what the heck? Like, I just slaved over that video. It turns out I could have just asked you for all the help. Yeah, very fun, very fun. Sorry, I cut you off, Resta. I was just excited to hear more about unculturable microbes.
there are definitely some cool bugs out there.
All right. So today we are going to be talking about bioinformatics, but instead of doing it like a lecture, again, we're going to walk you through, or not me, I'm actually going to enjoy learning alongside of you because, again, I failed this class, so it's going to be interesting to revisit some of this material. But we're actually going to set up an environment where chat can actually participate and sort of figure out how someone with a computer science background or a programming background can tackle biological problems in a way that you could call vanilla biologist like myself could not. So unique problems that have unique solutions is something that I'm really excited for. So we are going to specifically take a look at how we can harness the power of programming to defeat something like cholera. So cholera is a particularly nasty pathogen, which I'll talk a little bit about in the future. But this angle that you can approach drug design with is something that can only be done with the help of a computer. And so if you can wield that sort of power, you can contribute quite a bit to our understanding of biology. Bioinformatics is genuinely so fun once you get over the initial learning curve as a biologist. I know. I enjoyed the class. What filtered me out wasn't how to think like a bioinformaticist, but rather I'm just bad at programming. Like, I would write down the steps correctly and be like, oh, this is how I would algorithmically approach this problem, but I'm just like bad at typing the types, you know? Okay, so let's hear about cholera. So Vibrio cholerae is the pathogen that gives you cholera. It's very unique and an interesting pathogen on its own. I actually considered making a video solely on how interesting cholera is, but at this point, the list of ideas that I have to make content with it exceeds my capacity. You know, I do die every 8 days, but I think my entire cell lineage will die before I finish covering everything that I want to cover. So we'll give you a little tease here, and then I'll decide whether or not I still want to make a big cholera video in the future. But for now, let's talk a little bit about why it's pressing that we find a solution to this cholera issue. So cholera is an infection that is spread by ingestion of contaminated water. The resultant infection causes diarrhea, causing severe dehydration and ionic imbalance. So untreated, cholera can actually kill in the span of hours. So this is actually something that is quite terrifying. I've talked about tuberculosis on stream before as being like an S-tier scary pathogen. Cholera is also pretty scary just because you can go from feeling relatively okay to dead in a very, very short amount of time. According to the World Health Organization, diarrheal diseases are the second leading cause of death for children under the age of 5. So this is, you know, a pretty big issue globally. And as of 2002, so it's a little old at this point in terms of the data, but just to give you an idea of how shockingly dangerous cholera can be, the case fatality ratio was 3.95%. So that is pretty severe. There is a decent chance that you are severely affected by a cholera infection. Although it isn't apocalyptic, people do get better, but it is prevalent, we'll say. What does that percentage mean? I believe case fatality. I'm not prepared to super talk about that because I'm not like a medical person, but I do believe that is correct. That like there's you know, there will be people that recover from it with intervention, but if you don't have intervention, that can be pretty severe. So this is the part that, as a molecular and cell biologist, the part about cholera that I find to be interesting, as well as sort of the mechanism of action. So how cholera does what it does is sort of my entry point into this problem. And when Resta pitched this idea, oh, by the way, if you're from Resta's audience and you haven't seen me fawn over how hard Resta has worked for this collab, I gave her the opportunity to do a journal club that I would mainly guide, but Resta wanted to take this 101 series by the horns, which I greatly appreciate because, again, without her discipline, this would not happen. I would have picked one paper and we would have talked about it instead of this more interesting, I think, form of engagement. And so she had picked this problem, and I thought, oh, that's awesome because I'm already thinking about cholera for maybe some future content. And so this is what I've cobbled together in terms of my preliminary research into cholera. So cholera makes this toxin. It's the cholera toxin. And what it does is that it jams the internal signaling pathways of your intestinal cells. So unlike other toxins that go in and just kill the cell, this particular toxin kind of will gum up the internal signaling pathways and cause a buildup of this signaler called cyclic AMP. And what the cyclic AMP buildup will do inside the cell is it'll actually power some of the pumps that maintain the osmotic pressure in your cells where they need to go, where they need to be, right? So your cells are constantly actively putting in the energy to pump out water when they need to, or to pump out different ions when they need to. And this tricky toxin will cause a buildup of the signal that says pump everything out. So instead of killing the cell outright and releasing all of its contents, it'll actually cause the cell to spit out bicarbonate, water, sodium, potassium into the GI cavity. Now, when your lining of the cells that make up your GI tract, when they are told to expunge everything in their bodies, that's going to cause an excess buildup of fluid and ions, and that is what is going to cause diarrhea. And so when you lose that much water and ions in a very short amount of time, that can cause your body to go into shock. It was correct. This is not a pleasant way to go. Not that there is a pleasant way to go, but you can imagine that this particular pathogen causes a lot of human suffering. And so being able to think about ways to tackle this problem is going to be worthwhile, worth doing. Just as an appreciation for molecular biology, although this cholera toxin is objectively horrible, it is somewhat aesthetic. It's what's known as a pentamer, so a protein that has 5 identical subunits, which have this satisfying symmetry. Do the cells die as they're evacuating all their fluid? I don't know that for a fact, but I imagine that when they are that like imbalanced, that is going to cause some amount of cell death. Probably, I'd say probably, although I haven't read those exact words, more juice. So something that really, really interests me about cholera from like a biology evolution story perspective is the fact that Vibrio itself is not the thing that actually causes this cholera toxin. You can be infected by Vibrio cholerae and be totally fine. The thing that actually hurts you is the bacteriophage that infects the Vibrio bacteria. So the thing that makes the toxin isn't the bacteria, it's the phage that infects that bacteria. So this set of pathogens have evolved a very interesting evolutionary relationship with one another, where the cholera, the Vibrio cholerae, will provide shelter and a means of protein synthesis for the bacteriophage, and the bacteriophage in return provide a way for Vibrio to spread back into the environment, right? So I've talked over and over again on this channel about the fact that you don't win the game of evolution by being the strongest or fastest or whatever. You win the evolution game by spreading your genes, and some strategies involve this antagonistic relationship with their megahost, right? So even though the host for this bacteriophage is technically Vibrio cholerae, the ultimate host, the mega host, is the the human, right, or the animal, whatever animal Vibrio can infect. What a nice relationship. It has been described in the papers that I've read as a complex and elegant interplay between host and phage. My ideal relationship. Interesting. I'll say interesting. And so this is what interests me about cholera. But the thing is this is not what we're going to be talking about today. We are going to be talking about how to stop this from hurting you. The dehydration and the electrolyte loss probably puts the entire body into ginger mode. Exactly. It puts them into shock. So great progress has been made into treating cholera. First, infrastructure is extremely important. So never ever want to downplay the work that non-scientists do when it comes to mitigating the effects of horrible diseases, right? A lot of improved infrastructure means that you can have improved sanitation practices. Improved treatment exists. So you have a pretty good chance at surviving a cholera infection if you are rehydrated very soon after symptoms start showing. And medicines. So several vaccines have been made. Treatments, antibiotics exist for treatment of cholera infection. But just because we have the stuff to treat cholera doesn't necessarily make it not a problem, right? Cholera remains an issue worldwide. And whenever we talk about using antibiotics for the treatment of bacteria, we always have to be cognizant of the fact that antibiotic resistance can and is evolving. Also vaccine escape as well. Does an increased sanitation improve your chances of survival, or does it help the spread? So improved sanitation, I believe, will primarily prevent cholera from jumping from person to person. Okay. So here is the first bit where chat can flex your big brains. Let's say that we are coming up with a strategy to fight antibiotic resistance in the cholera phage, or the cholera pathogen. How would you target the cholera bacteria? I want to hear some answers from chat. Knowing that stuff like streptomycin can have that relying on antibiotics like streptomycin can cause drug resistance in cholera. So how would we fight this bacteria back? Icosahedron says phage therapy. That is one approach that we can certainly take, and that's definitely a very hot topic recently about using bacteriophage to specifically target bacteria. That is true. That is one potential strategy. A says fight the bacteriophage. That is also a great strategy. Deploy nanomachines. That might not even be that far off from... We might not be too far away from a future where that can happen. That is a very interesting strategy as well. More juice. So these are pretty good. What else could we do? So we can fight the bacteriophage. We can fight it using phage. Try to blow the bacteria up. What are some other ways? Some other creative ways that we can get around using antibiotics like streptomycin, ampicillin? Does the easy answer of antibiotics count? We'll say with an asterisk. So currently, right now, a lot of the antibiotics that are used against bacteria will in some way destroy the membrane or pop the bacteria in some sort of fashion. It's not all of the antibiotics that exist, but certainly a good number of them are membrane busters, we'll call them. Knowledge proliferation. Well, in an indirect way, if some of you are biologists or budding biologists, this can help spark some interest and creativity. Gene therapy, that is interesting, Brandon, in terms of what you mean by that, exactly how gene therapy can play a role in fighting cholera. Nanobodies is certainly an interesting approach as well, although I am not entirely sure how well nanobodies work in a hostile environment like the GI tract. I'm not saying that nanobodies is a bad answer. I'm just saying that I don't really know. Morjuice asks, how is cholera becoming antibiotically resistant? So generally, bacteria will evolve or mutate, not on purpose, of course, but mutations will happen that will cause the things that the antibiotics need to bind to vary very slightly. Something targeting their pump secretion systems, that's also a very good answer, Kit. So we can design or find antibiotics that work in a slightly different way, or in a different enough way that doesn't have to do with the ways that are commonly used. So it's a bit of a roundabout way of saying, let's try to find a new way to fight bacteria. I think these ideas all overlap in that way where if we don't want to rely on streptomycin simply, we have to find something else, right? Something that doesn't do what streptomycin does, a new class of antibiotics. Now, some of you in chat might be thinking, well, if we use antibiotics, isn't that just going to push the problem down the line? And the answer is, yeah, kind of. That's why this cooperative effect of improved infrastructure, improved sanitation, improved treatment, and improved antibiotics, it's not just a fight on one front, right? To prevent antibiotics from, making bacteria super resistant to one class of drug, we might need to develop another. And will this push the thing back? Will we ever be rid of this problem of drug resistance? That's a really hard question to answer. But what is true is that species are extremely resistant to being eliminated, right? Evolution has nearly perfected the art of survival, especially amongst microbes like cholera, right? So the dream of eliminating cholera worldwide, I'm not saying it's impossible. I'm saying cholera will not go down quietly into the night, right? Yeah. We need find another kind of marker to identify the bacteria I'm not entirely sure which of those strategies that applies to, but basically your answers have been quite good. But I'm going to summarize those answers by saying we need to find new ways to fight bacteria. So this is my list of tricks. And certainly this is a bit more of a generalist. I was trying to capture very general approaches, right? So 1, find/design a drug that shuts down an essential metabolic pathway in Vibrio cholerae. So some of you have recommended something that looks like this. Or find/design a drug that shuts down production of the cholera toxin. So I forgot who it was who had said it earlier in chat, but targeting the bacteriophage that makes a toxin certainly a very, very viable strategy. Both of these answers, 2 and 3, both are around that idea of inhibiting the phage that makes cholera bacteria dangerous. But today we're actually going to go with this 4th approach, right? Find and design a drug that shuts down replication of Vibrio. So again, the class of drugs that we use to treat cholera now and that cholera is becoming resistant to are membrane busters. So what if we had a way to prevent that bacteria from replicating? That's the angle that we are going to approach this problem with today. And here is where I will now transition to Resta's portion of this presentation. So how are we going to shut down the ability for Vibrio to replicate, Resta? Let's get into the part where I become the student.
Well, in addressing that, we're actually going to take a clue from a journal article called Targeting the Bacterial Orisome in the Search for New Antibiotics. That's the journal up there. But first, let's define the orosome. The orosome is made up of multiple copies of an initiator protein, DNA A, that form an oligomer at the chromosomal replication origin called ORI-C. This oligomer causes a nearby AT-rich region to unwind. That is where you have lots of adenines and thymines. Once this region is unwound, a helicase called DNA B and other proteins will be recruited to that site to form the bacterial replisome, which carries out DNA replication. As you can see here, we have DNA A, and so there you can see ORI-C, and in the top right, you can see multiple DNA A's forming that oligomer, and how that unwinds that bit of DNA in the loaded section, and that leads to the completion of the replisome down at the bottom there. So, if a drug can be designed to disrupt initiation of DNA replication by DNA A in a way that is lethal to Vibrio cholera, it could serve as a means to combat cholera infections.
Yeah, so we got a question, we got an answer from Mandrake here. So the idea, I assume, is to make an RNA that binds to the replication machinery. That would be one of the goals that could certainly fit this strategy. Yeah. So you can go on, Resta.
Okay. So, whoa, whoa. So, in this line of research, it could be important to get a picture of where DNA binds within the Vibrio cholera genome. The regions where DNA binds are called DNA boxes. You can see them over here as yellow triangles. As multiple DNA proteins are required to form the aforementioned oligomer, ORC regions contain multiple DNA boxes that are clustered together. And as you can see in this figure, the sequences of DNA boxes of many bacteria are known. But for the purposes of today's presentation, let's say we don't know what the DNA boxes within the Vibrio cholera genome are. In which case, chat. What are some approaches we could take to find the exact sequences of DNA boxes within the Vibrio cholera genome?
Yeah, this is sort of a great, interesting example. So for some of the biologists out there, and Resta, you can correct me if I'm wrong, but we can think of DNA boxes just as places where DNA likes to bind and initiate replication. Is that correct?
Yep.
And then so the goal here would be to try to find where DNA likes to bind. And so for the biologists in the chat, the reason why this is a compelling question is because, or I don't know, Resta, if you want to, if I'm spoiling what's about to come up, but I think I'm reiterating the confusion here, right? The fact that we don't know where these DNA boxes are, and the question would be how to find that. Is that accurate? AT-rich means part of the genome saturated with A and T. I believe that's correct, right, Resta? These AT-rich regions?
Yep.
Do you happen to know why there are AT... This is totally off the cuff, but do you know why that there are AT-rich regions before the DNA boxes? It seems sort of greater than chance. This is kind of interesting.
Not sure. Maybe it could be related to the number of hydrogen bonds that A and T have versus C and G. Who knows?
Yeah. So for mute, if I'm being honest, as a non-biology student, this went over my head a bit. So let me try, and Resta again can correct me if I'm wrong. To stop the bacteria from reproducing, our strategy here is going to be to find how it starts or where in the DNA it likes to... Okay, so let me back up. Before a bacteria can divide, it needs to first copy over its DNA, and it's got to make 2 copies of its DNA, and then it can split into 2, right? So in order to stop bacteria from reproducing, what we can do is try to find a way to shut down that DNA replication process from happening. So if we look more closely as to how the bacteria reproduce or replicate their DNA, they do so, and they have to have a starting point. This starting point is this DNA box, and we want to find where these boxes are. So if we can design something that stops it from starting to replicate, we can stop them from reproducing. Hopefully that makes a bit more sense. Some other questions are filing in. Answers are filing in. So Mordred says this might be a crazy guess, but we can sample a bunch of DNA that's going through replications. That is interesting. I wonder how you can tell if there's a way you can discriminate what because there are also different sites, right? It's not one site. The DNA boxes occur all over in the orosome, and so that might be kind of challenging to differentiate which DNA belongs where. Also, sequencing the DNA can have slight variations in terms of when the polymerases actually start. There's some wiggle room, at least in a practical setting, there's some wiggle room exactly where those things begin. So looking at the resultant DNA, I think it might work, but I think there are some issues there. That's a good suggestion, though. And then filter out the sections that have DNA bound somehow. whether or not you can pull down DNA, that's certainly an interesting way you could do that. I don't know how tightly DNA binds to the DNA boxes, whether or not they bind tight enough, because you can do that with chromatin, but I'm not entirely sure you can do that with DNA. It's a good idea though. That's really good. I wrote a simple algorithm that does a task that is essentially what you are describing. Ooh, Aiden, do not spoil it. You can chill here and vibe along, but if you know the answer, don't spoil it, because to give the non computation people a chance, and the computation people that don't know the answer a chance to work it out, is also interesting. I assume you need the whole genome sequence analyzed, and then you can start making oligo probes. Certainly true, but whether or how making oligo probes can directly lead to the discovery of these DNA boxes, I'm not entirely sure. But I think these are good hypotheses, good strategies. This is a fun thought exercise. It is a fun thought exercise, and we're gonna let people have their fun, because it genuinely is very interesting. Again, I didn't drop the class because I hated it. I dropped the class because I was bad at it. All right, Resta, do you think we're ready to move on?
Yep. Okay. So, addressing this question is where a bioinformatician's work begins. One thing we can do is redefine the biological question. What are the sequences of DNA boxes within the Vibrio cholera or C region into a series of problems that can be solved with the computer? The method we'll go over today actually comes from the textbook Bioinformatic Algorithms, which is the same textbook I learned this from. Definitely recommend if you're bioinformatics minded. But before we dive into that, I'd like to note that Vibrio cholera is a bit special in that it contains 2 circular chromosomes, each with a different origin of replication. Chromosome 1, shown in blue on the left, contains an ORC type origin, and chromosome 2, in orange there, contains a P1 plasmid type origin. Since replication of chromosome 2 works differently than the replication model introduced so far, we're going to focus our discussion on the DNA boxes present within chromosome 1's ORC region. So, that said, here we have the sequence of the ORC region in Vibrio cholera. It is 540 nucleotides long, and within it, we expect there to be multiple DNA boxes, again, so that the DNA protein can bind to multiple places and form that oligomer to initiate DNA replication. We know this is the ORC region based on experimental and bioinformatic evidence, which I won't get too much into. However, note that there are tools, like you can see on the right, called ORFinder, that are used to infer putative ORC regions, given the sequence of a circular bacterial chromosome. Yet, even with the ORC sequence, we still need a method to figure out which nucleotides belong to DNA boxes. And to answer this, we can start by abstracting the problem. So, let's take a step back. Since we can think of DNA boxes as biological words that tell DNA, in a sense, where to bind, we can think about how to find hidden messages in a string in the first place. So here, we have a pirate who wants to leave the following message in a bottle containing clues to a treasure's location. It says, "To find me treasure, heed these words and let no doubt cloud your mind. Sail to the isle where the moonlight dances and on the water round the crescent bay. There beneath the ancient oak with the branches wide, seek the X that marks the spot where shadows hide. When the sun sets and the stars are high, dig deep where the cold winds sigh.
That's awesome. That's awesome.
But let's say this pirate wants a bit more security than just writing that in plain English. Well, one of his options is to use a Caesar cipher, which replaces every letter with the letter in the alphabet that is 7 letters down. So, just starting from here, do you have any ideas, chat, of how we might recover the original message if what we're given instead of that bit of poetry is of so on?
For context, chat, I got to see these slides before because we were collaborating. I was like, this activity is so cool. Yeah, so this is actually interesting from like a D&D puzzle perspective, because I love doing Caesar ciphers as a dungeon master, and the first time that my players went through it, they actually did do exactly what the answer is to solve this. So it is very, I think, natural to come up with this solution. So imagine you didn't know what the shift was. So for those of you who say go back 7 letters back, let's pretend you had no idea what the shift was. I'm actually going to maybe hide the pirate half, see if I can do that. Yeah. So if you just got this message in a bottle and you had no idea what the shift was, but you knew that this was some sort of paragraph, how might you start to tackle this issue? So take a look at the chat. So frequency analysis, statistically determine which letter corresponds with which, and hey, it's just 7 down. Got it. You might not even know that this is a Caesar cipher. If you have a hypothesis that this is a Caesar cipher, then certainly trying to shift every letter by a certain amount is a natural way to start things going. Iterate through potential shifts and look for common words. Iteration is definitely going to be a part of this. A tool like binwalk that shows the most frequent segments. That is interesting. Frequency analysis probably needs way more than just way more data than a paragraph. Look for common words. Once you've determined E, the most common letter with the frequency analysis. So how do these answers sound to you, Resta?
They sound on the mark, especially looking for common words.
Yes. Very good, chat. Very good. See, it's and embarrassingly, when we were running through this for practice, I did not answer that. I did not answer correctly. So chat, your big brain, you hold one over me. Frequency analysis, looking for common words. Fantastic. All right, let's continue then.
Okie dokes. So, if we make the assumption that the message in a bottle is written in English, we can expect that the word the is present multiple times within the message. And since the most common 3-letter word encoded in this message is AOL, it's reasonable to guess that the letters A, O, and L correspond to the letters T, H, and E. And if we notice that the alphabetic distance between each letter pair is 7, then we could try to decode the full message by shifting all letters by 7. And since doing so produces a legible message, we can conclude that that's probably the message we're looking for. And so with this in mind, how might you go about looking for DNA boxes given all of this?
Yeah, so we'll let chat chew on that for a second. So the approach, chat has completely nailed. We're looking for frequent words because we have this assumption that if there is a frequent pattern, that it means something. And so how might we do that? Algorithmically, right, Resta? You mean to say algorithmically, how would you think about tackling this or implementing this solution?
Yep.
So I want to go back and answer some of the questions from chat. So Mute says score. So there are common patterns in DNA, RNA. So this, I think, is a very interesting computational question because let's say, and I'll type this out in chat, that you have a completely random string of DNA. Well, if that is the case, if you have a string of, let's say, five, a string of DNA that's 5 letters long, then how many different combinations could that be, right? It's 4 to the 5th. So there are so many different combinations of how letters can be randomly generated that if you have common repeating patterns, it doesn't necessarily mean that those are DNA boxes. But there is a chance that that higher than random chance sequence might have some sort of function. Dr. Toto says you look for common parts of the DNA, right? I'm not entirely sure what you mean by that. I know some DNA markers that are all the same in all humans. When we say marker, what do we mean exactly? More Juice says, oh, is there like a common set pattern of DNA boxes? I'm not like a bacteria geneticist, so I can't say that for a fact whether or not there are like how evolutionarily conserved DNA boxes are. Because when we were running through this with Resta, that was actually my first instinct as a molecular biologist. I was like, oh, well, I would look at Vibrio's cousin and see what Vibrio's cousin's ories look like. But I believe that they're not that similar between Vibrio cholerae and its nearest cousin. So that approach might not be as valid. Morjuice also says, run through the genome and look for common repeating substrings. Interesting thought. Factor analysis could be fun too. I don't know enough about statistics about to know whether or not that is true, Mylan. Although when you see the solution, maybe you can elaborate on what that would mean. You could look for more complicated things like machine learning to find clusters of code. Ooh, we don't need to go that far though, which is why I think this stuff is interesting. For me, thinking about machine learning and AI and stuff like that takes some of the, I don't know, human aspect of discovery out of it. So while I acknowledge that machine learning is in fact very important for the field of biology, I don't think I would have picked a machine learning problem, or I would have probably vetoed a machine learning problem because it's not quite as intellectually stimulating unless you're the one building the machine learning algorithm. In forensic science, there are some parts of the DNA sequence that are the same. Yes, exactly. So we're trying to look for sequences that might correspond to what we're looking for. However, the challenge with that, Toto, is that we don't know exactly what we're looking for. We just have this hypothesis that what we're looking for is probably repeated non-randomly or not likely to be random throughout that sequence. Look at the frequency of codons. Ooh. So we're not actually talking about codons here because the origin of replication doesn't have to do with making protein. So codons are not going to be a part of the conversation. What's the difference between molecular biology and biochemistry? Biochemistry is a subfield of molecular biology. It's included in that umbrella. Okay, so Resta, do you spot a correct answer in the previous answers that chat gave? Any of those sound good to you? If you want to peruse what that looks like. I think I see one that looks pretty close to what we're aiming for.
Me too.
Yeah.
Also, regarding that question you had earlier, certainly comparative analysis is an approach you could take, since DNA boxes of other bacteria are known. And in general, they are sequences of 9 nucleotides. And you could use that to try to find DNA boxes within Vibrio cholera. However, there's a chance that if the DNA boxes in Vibrio cholera are significantly different from our references, then we might miss some.
I think that's the wonderful thing about science is that your thoughts in chat, there are a lot of good ones out there, but the beautiful thing about science is that there's a tool for every question and you can debate what the best tool is, but there's a chance that actually if you use all of the tools, it's better than using just one. So looking at things like what Toto was suggesting, like looking at commonalities, like I had suggested with looking at cholera's cousin to see if there's some conserved element about DNA boxes. That's one approach. And then someone else may have a completely different approach. They both might yield answers and perspectives that the other approach did not have. So certainly that is one way to do it. It is a more hypothesis-driven approach, and this is a less hypothesis-driven approach, right? Like with the conservation, our hypothesis is that Vibrio is likely like its cousins, but this is a more naive approach, and so it's interesting to think about what the pros and cons. Actually, I'll ask this question to you, chat. What are the pros and cons of a hypothesis-driven approach, like looking at cholera's cousins, and a more agnostic approach, like looking for common words in the Ori? Missing some is better than missing all. That is true for sure. Knowing DNA boxes are 9 sequences long would most certainly simplify the search process quite a bit. That is very true. Very true. Yeah, so I talk with my students actually quite a bit in terms of helping cultivate their scientific sense. So we have the hypothesis-driven approach of let's look at cholera's cousins, and we have the naive approach, which is let's just look for common words. Let's not assume that Vibrio cholera is anything like its cousins. What are the pros and cons of both? Hypothesis-driven approaches are great if you're on target, but they rely on what we currently know. That's a great answer, Mylan. I guess one could be more biased. Exactly. Bias isn't necessarily bad if you're right. If you don't get punished. If you're off base with your initial hypothesis, you might end up getting nowhere. Exactly. Like it's more targeted. But what are the benefits, right? So of course, in our bioinformatics stream, we want to sell you this unbiased approach. But what are the benefits of a biased approach? Because there certainly are. In fact, if you were to ask me to tackle this problem, I personally would have looked to cholera's cousins and taken that approach just from my school of thought, right? I'm a very, very molecular person and not very much a computer person at all. So I don't want to undersell the approach of looking at cholera's cousins. It's just not perfect, much like any approach in science isn't perfect. Benefits are that it guides your search. It helps you fit into your current theory, allows you to find pre-existing literature. Exactly. It's always tempting, I think, for early scientists to always want to go for an unbiased approach because they are afraid of not covering everything, when in reality, it's not a one-size-fits-all approach. You just have to take the advice of people that know better than you in your field as to what is more likely to work. Because I'm not saying I'm right or Resta's right, because we had two different approaches, right? Because I'm not an experienced cholera researcher, nor am I a bacterial geneticist. I'm here acknowledging that we have schools of thought that we're trained in, and our schools of thought are good at different things, and they might complement each other. One might be better than the other, but it kind of depends. And whether or not you decide an unbiased approach versus a biased approach is appropriate kind of depends on like the art of science. This is the art, right? Like people think science is so like, this is true, this is false. It's more like we are out here and we are trying to do a very difficult thing, which is to uncover the mysteries of the universe. And there's no one that can tell us what the right approach is. We just have to try, right? And I think that's part of what's so exciting about doing science. It also lets you save resources. That is a non-trivial advantage. As long as you can recognize you are biased, you can gauge this effect on that bias. That is true. And it's something that I encourage my students to talk about when they write their lab reports, is that if you have a bias in your approach, you should talk about that so that you don't get roasted by somebody else. If you roast yourself before someone roasts you, you're considered responsible. If you don't, then you're considered neglectful and a bad scientist. Saving resources is a big deal, considering what good is science if only the high-powered labs can do it. So if you are a smaller lab with less money, you might be guided towards these more biased approaches that allow you to work with fewer resources. RPing reviewer 2 is a very valuable skill. That is true. Okay, great. I love this conversation. It's so good. And hopefully you are getting something out of how we can untangle the mystery behind how science works. Although it is worth noting out that right now I do see a couple of scientists in my chat right now. So it's a little cheating. But I'm glad to have the participation. Okay, Resta, I think we're ready to go into how we are actually going to implement this solution of finding common words in an algorithmic way.
Actually, before that, do you
Yes.
mind if I go further down this tangent?
Yes,
'Cause
please, please.
it's definitely a very, very interesting point. Well, some examples from bioinformatics that show like when you might want to use a reference versus when a more naive approach might produce more optimal results include, for example, uh, reassembling the genomes of extinct animals. For example, there's been some buzz going around about bringing mammoths back. Technically, they won't be mammoths, they will be elephants, modern elephants, hybridized with mammoth DNA. However, in the process of constructing the original mammoth genome, they referenced the genomes of elephants and related organisms to kind of fill in the gaps. Um, so you can do some, when you simply just don't have that information available, having a reference is invaluable. In addition, on the flip side, uh, there is a field called metagenomics that seeks to discover all the microbes in a particular environment. Now, typically, the most cost-effective approach of surveying the population of microbes in a sample would be using an amplicon-based method. I won't go too much into that, but essentially you use a kind of barcode, either 16S or 18S region, and there's databases saying like, oh, this 16S sequence comes from this organism. 16S, it's typically used in prokaryotes, and 18S is used in eukaryotes.
And those are the ribosomal RNAs, right? Just to
Yes.
yeah. So looking at a part that is unique to their I get maybe family.
Yep.
Yeah.
But the thing is, the majority of microbes that are out there are unculturable. We don't have genome sequences for them. We don't have like physiological information about them. It's this huge number of microbes that we know little to nothing about is often called microbial dark matter. And and if you want to study that, you got to take a naive approach. That is, sequence all the DNA within a sample and then try to reconstruct genomes from that, which is a pretty incredible thing to do because samples will have many, many different kinds of microbes. And you want to make sure that the pieces that you're using to reassemble the genome go into the right genome.
Yeah, I actually dropped out of my bioinformatics class at around this point because this process is like assembling a puzzle from hell, right? Think about when I did my video about archaea, right? Archaea in the gut. The way that they found archaea in the gut wasn't, oh, they cultured the archaea out of the gut, but they took a swab of the gut and all of the DNA from not only your cells that slough off into the gut environment, but also all the yeast, all the bacteria, all the phages that all have DNA, all of that DNA gets like blended up together. And somehow, with the power of computers that is far beyond my like understanding, you are able to separate that like DNA soup into different puzzle pieces that can be reassembled. And the challenge of doing that is making sure, again, as Resta said, that the right pieces go in the right places. It's just incredible. And again, not something you can do by hand, which is why we'll let the bioinformaticists do the majority of that work. Very cool. Very cool. I love that kind of stuff. Biological dark matter.
And,
It's so cool.
Okay, I was going to say something, but I shouldn't say it because it's doxing. But yes, it is cool.
All right.
Anywho, So far, we've shown a way how to find hidden messages within English text using the highly frequent word the. But if we want to apply this to Vibrio cholerae or a C region, we need to define what biological words are. So, within the pirate message, we had the advantage of spaces between words. This informed us of how long each word is. However, when working with DNA, we don't have this advantage. So instead, we need to check every possible length of a word to identify which words could potentially be DNA boxes. To do this, we will use k-mers, where it's a sequence of arbitrary length k. For example, the figure on the right over there shows all possible formers of that sequence in yellow up there. You could make a similar diagram showing possible 3-mers, 5-mers, and so on. So, remember how I mentioned that bioinformaticians redefine biological problems as computational ones? Well, to address the question, what are the sequences of DNA boxes within Vibrio cholerae or C region, we can formalize the following questions. The first of which is, what are all the possible k-mers in the Vibrio cholerae or C region? We can do this by sliding a window of length k across the Vibrio cholerae or C region, as you can see here. Just moving it one nucleotide at a time.
Can we see that animation one more time, just in case?
Sure thing.
Yeah. So this is the part that I found to be really interesting about my bioinformatics classes, because, again, I dropped the class because I sucked at programming, but this part actually does make a lot of sense to me, right? Like, if you're looking for common words and you don't have the advantage of English where you have spaces in between words, how would you do it normally as a human? It would be very laborious, but you just scan with these windows, and I was like, this makes a lot of intuitive sense. And I think if you are like a burgeoning biology student and you find this kind of like puzzle solving to be appealing, I highly recommend that you go and take some programming classes alongside with your biology classes, just to help develop you into this kind of puzzle solver, because I would not last 10 seconds in Resta's realm, because I'm not fit for that kind of thinking. I like more narrative storytelling kind of approaches. But if this thing kind of tickles your brain, I really do think that this is really cool. It's not for me, but I do see the appeal. Where I'm like, oh yeah, you can slide the window down. That makes perfect sense. It's really neat.
Also in chat, I noticed that Mute said, oh no, sounds like O equals N. So a bit of a tangent, but I found that a bit funny, because certainly with this small OEC region, that is like 480 nucleotides, you can do this. It's not going to take all too much computational power. However, when you're working with multiple genomes, huge amounts of data, terabytes upon terabytes upon terabytes, you really then have to start thinking about how to make your algorithms more efficient. And one way to measure that, in a sense, is using big O notation, which describes how quickly the problem but essentially you don't want your program to take exponentially large amounts of time in relation to the size of the input data.
Yeah, I'm getting flashbacks to Python, sort, quick sort, BOGO sort, or all these things are popping back into my memory. So it's cool to call back to big O notation. Very neat, very neat. All right. Shall we continue?
Yep. Okay. Let's see. Once we've done that, and we have a collection of all possible k-mers in the Vibrio cholerae or C region, we can then ask, what are the most frequent k-mers for each length k in the Vibrio Cholera Orisome region? By calculating this for k-mers of length 3 and above, we can produce this table here. Now, chat, does anything stand out to you about this table?
And chat, if you get this, you will have done better than me, because when we were practicing, I was like, I don't know, I'm kind of stupid. I don't really see it. But hopefully chat will do better than me. This is one of those things that you have to kind of zoom in for. I love k-mer analysis on the genome scale. I could finally practice using my CPU. That's so funny. Yeah, I enjoyed all the k-mer stuff. Unfortunately, that's when my alignment programs just started blowing up on me, and then I was like, man. I'm gonna go back and chat, and while you're thinking about if there's anything funny or I shouldn't say funny, but noteworthy about the k-mers shown on screen, I'm gonna go back and talk about some of the questions that chat messages that I'd seen earlier. Kelly Rogers had said, I'm not an expert by any means, but could phages be designed to specifically target cholera? So it depends on whether or not there is a conserved region or a conserved protein that a phage would like to bind to that only targets cholera, and then being able to package something that would destroy that cholera and have it be something that is specific to cholera. I'd say that as a general approach, this is very hot right now in terms of the field of designing new next generation antibiotics. The idea of using phage to destroy bacteria is very hot. So I'll say that it is theoretically possible. Whether or not it is practically possible is actually a different story. That might be a good thing for me to cover in a future stream. I don't know the answer immediately, but it is an interesting strategy to use. So I'm saying Morju says there's quite a bit of overlap, 5 mers and 6 mers, for example. Same with 7 and 8. Watching on 444p since I'm traveling, sad. Yeah, the font is a little small on your phone, I suppose. There's only one k-mer that doesn't contain TGA. That's interesting. All right. So some good answers in here. 4th, 5th, 7th, 8th, and 9th length k-mers begin the same. Okay, so I think we're hovering over a similar answer. Resta, what do you think about those answers?
Certainly getting warmer.
Yeah, that part I got too, in terms of there's some overlap, but then that's all I got. There's something trickier, certainly. Resta, would you say that if you have a formal education in biology, that it's easier to see what answer you're fishing for? Or am I thinking of a different part of the presentation?
I think so, simply because like you have the nucleotide complements like by heart, and
I think you kind of
can identify certain things.
Gave a little bit of a hint to chat right there, maybe unknowingly. Yeah. What if we opened that idea to the chat? Is there anything that you can recognize with nucleotide complements? So I'll go ahead and write the pairing. So G pairs with C, A pairs with T. The way I remember this is GCAT. I just think that's an easier way to remember it. So G pairs with C, A pairs with T. And if that information helps, there's something curious that I missed when I was looking at these strings. Morji says, are we talking about palindromes? I actually don't remember the answer. I remember approximately what the answer is, but palindromes are something that happens a lot in DNA stuff as well. All right, I think chat's had enough time pondering. So what's unusual about this set of k-mers?
Well, if you do the math, you'll notice that the probability of a 9-mer appearing 3 or more times in a randomly generated DNA string of like length 500 is approximately 1 in 1,300. So remember back to the pirate example where it would be highly unlikely that in a paragraph of like 200 words, that if you just chose all the letters randomly, that you'd get T, H, and E all together as many times as you would in the English language. Well, similarly, here we've identified some of those potential useful words, but this time biological words, air quotes. And again, keep in mind the nucleotide pairings in the top right. But now I want to ask, since now that I've highlighted the 9mers, do you notice anything peculiar about the sequences of those nucleotide strings?
Mika Craft says, oh, the first 2 9mers are complementary.
Oh?
Opposite and reversed. Interesting. Yeah. Yeah, Mega Craft, you got that. Having 3 of a 9mer show up in a small 500 nucleotide sequence is highly unusual. Yeah, I mean, I think that kind of appeal is really exciting, just because, you know, you're thinking about probabilities, and when you find something that is unlikely, that's almost finding, like, I don't know, like candy underneath your pillow or something. It's like, ooh, found something unexpected. And the fact that there are a whole bunch of 9mers, it's an over-representation of an unusually high count for something. So, you know, statistically speaking, that might be something interesting to look at just from the probability perspective. Something to do with a string presupposing a longer string, so substrings. A says one codon keeps shifting to the right. Interesting.
Well, I'll continue on because you got it, chat. So, here we can see that there's 2 main 9mers, even though 4 are listed. There's ATG, ATCAAG at the top, and then CTTG, ATCAT. The second of which, as A mentioned, is shifted just a bit, but it's essentially part of the same word, as in the location the location of it within the ORC region is the same. Secondly, the first 9mer and the second 9mer are reverse complements of one another. So we haven't given you enough clues to put all the pieces together. So in the next slide, we're going to go over why a reverse complement might be significant, and by looking at DNA replication of circular chromosomes.
So Morjuice has an interesting question, and I'm actually curious too. So he says, how come the CTTGATC doesn't show up in the 7mer category?
Say that again?
Why doesn't the CTTGATC, the common in the 9mer, why doesn't that show up in the 7mer category?
I think it's because ATG, ATCA is a more frequent 7mer.
Oh, interesting. So this is a little filtered, maybe.
Okay,
that's interesting.
Yep. But that said, finding 9mers that occur 3 times each is much less likely to happen by chance than a 7mer that appears 5 times.
That is true. I suppose whatever algorithm spit this out maybe didn't consider that to be significant, just because it's not that crazy for that to appear 3 times as a 7mer. I think the 9mer column shows that it shows up 3 times because the count is 3.
Yep, each of those 9mers shows up 3 times.
Yep.
And certainly more juice. You'd think that maybe that CTTGATC would appear 9 times. However, as you'll see in a bit, it's because the second, third, and fourth 9mers are all the same locations.
All right, interesting.
Anywho, before that, recall that the ribosome carries out DNA replication, as you can see there. In the left figure, you can see that DNA replication occurs bidirectionally to produce 2 double-stranded daughter chromosomes. And in the right figure, you can see that during DNA replication, nucleotides are added in the 5' to 3' direction. So if you had a loci with nucleotides AAA, CCC in the 5' direction, its reverse complement would be GGG, TTT. So moving on from this, we can develop a hypothesis that the 2 main 9mers, ATGATCAAG, and its reverse complement CTTGATCAT, could both be binding sites for DNA A, as it's highly unlikely for 9mers to appear at this frequency. So, chat. It looks like we may have identified the DNA boxes in Vibrio cholera's or C region. However, we gotta be careful and consider other possibilities. Can you guys think of any alternative hypotheses for why these 2 9mers appear so frequently?
Yeah, so, sorry, Resta, could you say the question one more time?
Oh, sure thing. Can you guys think of any alternative hypotheses for why ATGATCAAG and CTTGATCAT appear so frequently?
Gotcha. So I'll say just in chat, just for people coming in, these sequences are, in fact, DNA A boxes, and we've done our search. Yay. Hypothesis 2. That's for chat to think about. I think this is really compelling. When we last rehearsed, I actually didn't quite understand why the reverse complement was significant, but I think today it clicked for me because you have bidirectional replication. If you see a 9mer in one direction and its reverse complement, it's like, I wonder why the reverse complement also shows up and it looks like in the same proportion as the other sequences. That actually is quite compelling. And also, what I'm noticing just in the highlighted sequences, Resta, is that the spacing between the blue and the orange is quite different. It looks like, and again, I'm not a bacterial geneticist, so I actually don't know the answer to this, but it looks like that the initiation of replication can be slightly different on either strand. I guess that kind of makes sense. You don't want things bumping into one another. Is it like a start and end sequence? One side would bind to its complement on the other side. That is true because it is double-sided, double-stranded DNA. But this is the representation of a single string of DNA. So in the same ladder, you have the canonical sequence and its reverse complement on the same strand. And so you have 2 equal strands. So it's like the replication machinery can pick and choose depending on which direction it's going to. I think that's very cool. Kind of a shot in the dark. They could be regulatory parts. Ooh, so that's kind of interesting, Freak. That's kind of interesting. Does the fact that this is bacterial DNA circular matter? I don't know. That is a good question that I don't have the brain APM to figure out knowing how I know bacterial replication works. It's been a while, but DNA can't be read backwards, can it? There is directionality in which DNA is read. The fact that they are reverse complement means that you read in the same direction with regards to 5 prime 3 prime. So it'll have some level of symmetry, if that kind of makes sense. Is that so replication is easier for the complementary strand? Yeah, so that would be where I was heading with that idea, is that like, because you have the reverse complement on the other side, you can kind of pick and choose. And again, Resta, you can correct me if I'm wrong. To answer the question that I posed as to what other hypothesis this can be, Meat says random chance, and we live in a simulation, sorry. I like that. I like that a lot. Stability of the bacterial plasmid. That's a little... it's a little imprecise, and that's not like, it sounds rude, but this is a language I use with my students, right? If they give me an answer and I want them to clarify, the correct scientific thing to say is that that language is a little imprecise. I'm not entirely sure what you mean by stability. Not to say that you're wrong, but again, being a little more precise with the language might help with evaluating whether or not that's true. The diagrams earlier, the replication machinery does start on both strands simultaneously. Yeah, that's what I believe as well. So it definitely is both stranded, right, Resta? I don't know whether or not it's exactly simultaneous. Again, biology is a spectrum, so whether or not it is exactly simultaneous, I'm not entirely sure. But you do get two directions. So you do get replication in both directions. So having the other reverse complement will allow the specificity you need to replicate on that other side. Okay, so Resta, do we have an answer in the chat that is correct in terms of an alternate hypothesis here?
Not the answer I'm looking for.
It's the answer that I was looking for. So I actually don't know what you're looking for, which is very exciting because we haven't gotten to this in rehearsal. So because I thought Mute nailed it. For me, hypothesis 2 was random chance and we live in a simulation. So the fact that I'm about to get this wrong is very exciting for me. So Resta, what's going on?
Well, this can arise if you see these sequences elsewhere in the genome, because if there are, in fact, DNA A boxes, we'd expect them to just be within the ORC region. However, there are things called transposable elements that can put repeats in multiple places in the genome, just kind of randomly. And so, to double check that these 9 mers, which we found, aren't just something that you find everywhere in the genome, we'll look at the entire genome. And for example, let's formalize the question. What are the indices of the 9-mer ATG8TCAA within the Vibrio cholera genome? And you can see on the right side that we in fact find that specific 9-mer 17 times. However, what gives us confidence that this 9-mer is a DNA A box is that only occurrences within the ORC region are clustered together. Again, because the DNA A proteins need to be within close proximity to one another to form the oligomers.
Okay, so let me process this out loud, Resta. So the alternate hypothesis that you have suggested is that maybe you can find this particular sequence by accident in the OREC because it jumped there, right? I just want to make sure the chat understands what a transposon is. A transposon is a sequence of DNA that can actually loop itself out of the DNA and somewhat randomly insert itself in a different place. If you want some nightmare fuel, your DNA can do this all the time. It's not just a bacterial thing, but you also have a significant portion of your DNA that can just pop out and then pop back in. That's insane to me. And the reason why we're not, pumped to the gills with cancer is because this can end up causing cancer. But the reason why you're not like human beings in general are not like pumped to the gills of cancer is because your immune system is very, very good at keeping those cells at bay or regulating their existence, we should say. So transposons are an incredibly interesting bit of biology. I actually had no idea that was coming. So that's actually really cool. So the alternate hypothesis is maybe they look like DNA boxes because they have this that they're nimers, they're unlikely. It's actually really interesting to think about the fact that they could have just popped in there because of a transposon. And so if I'm saying this correctly, and Resta, you can correct me, the reason why we think that they're still DNA boxes is because they're unnaturally close to one another within the ORAC. So these nimers exist outside of the bacteria chromosome. That is true. But actually, if we look at the spacing, the first two are 30,000 base pairs away. That's a huge distance. And then this cluster that you've identified, they're within what? They're dozens, maybe hundreds of base pairs away, it looks like, just eyeballing. Yeah, dozens to hundreds. And then we go to the right, and that's another 30,000 base pair jump. Those distances are really significant. They're really far apart, and we have this unnatural little cluster. And I'm wondering if you can calculate, not that I'm asking you to, but I'm wondering if it's something that people think about, like calculating whether or not this particular clustering is unusual. Very
There's
cool.
certainly metrics for that kind of stuff.
Oh, I love that. That's really, really interesting.
Quantitative way of saying, oh, this is a cluster versus, oh, this isn't a cluster.
That is so cool. So it's okay. So we're still not at the stage. So my background is in cell and molecular biology. So every time I read a computation paper, I just want to yell, we gotta do it in the lab. Like, you can't know until we do it in the lab. But I can see why this would be good justification to try this in the lab. That's very, very interesting. All right. Anything you wanted to add before continuing, Resta?
Well, at this point, we can be very confident that the two nimers we found are indeed DNA A boxes, at least putative ones, because you always have to take that grain of salt when doing bioinformatics. However, at least now we've taken our first step to potentially developing a drug for cholera infections, which we then follow up this with wet lab stuff as opposed to the dry lab stuff we did here. And I just want to rehash as well that earlier we redefined the biological questions. What are the sequences of DNA boxes within Vibrio cholera's or C region as computational questions? That is, what are all the possible k-mers in the Vibrio cholera or C region? What are the most frequent k-mers for each length k in the Vibrio cholera or C region? And what are the indices of the nimer ATG-ATCAA within the Vibrio cholera genome? And in practice, answering these computational questions requires computer code. Hence, coding is one of the key skills of a bioinformatician. And of course, there is more to bioinformatics than just coding, and I can talk a bit about it, maybe. But for now, let's wrap things up with a bit of Q&A.
Yeah, so could you, while we have chat, maybe ask some Q&A questions, maybe walk through what the pseudocode says? If it's too complicated, we can always leave that. But just for the younger students in chat, maybe who haven't had a programming class, is there a simple way to describe what this pseudocode actually does? Like line by line.
Okay, let's start at the left, where we're calculating all the possible k-mers within a given string. We have this thing called frequency table, which in computer science we would call a function because it's a bit of code that does a specific thing. and it takes as input a, a bit of text, which in computer science you'd call a string, as well as and some integer k, which would be like the max Kamer size to consider. And so first you instantiate this probably list called frequency map. So essentially, if you think about your computer and your RAM, you have to store information in some way. And the way that's done well, I'm oversimplifying, but you essentially store data in data structures, which in turn are well, yeah, at a higher level of abstraction. That's what you store the data in. At a lower level, that is closer to machine code, it's all the ones and zeros that get flipped. But and then you'd store the nucleotide sequence in a data structure. In this case, we're naming it N. It would be a string in Python, for example. And then you have a for loop where whenever you want to do something multiple times, you'll want to iterate over and over and over. You use this syntax, which very commonly across programming languages starts with the for keyword, in which case we start at I, an index, which is set to 0, and we continue down. We continue to iterate until we reach an index that is the value of the maximum Kamer that we want to consider, subtracted from the length of the text, or the length of the nucleotide sequence. Then, so then we take that Kamer, in this case, text within parentheses I, comma K, that would be the Kamer. We store that in pattern. It could be a list of strings, probably.
So is the pseudocode on the left here basically the stepping down, like the window sliding? Is that what that part is? Oh, I see. I have a question about Big O, actually, in terms of like other ways that you could design this, and I might get yelled at from the programming chatters, but how much worse would it be to generate every Kamer of K length and then check if that is a substring of the code? Is that crazy to do? Is that a lot worse than the window style? I'm just kind of curious.
Check if it's a substring of the code, you said?
Yeah, so let's say if I have a list of every possible former, and then I check the big string to see if any of the formers I generated are in that string. You're crazy Fi. I kind of want to know how much worse that is as an approach, just to show off the elegance of programming, right? Because this sliding window thing is pretty elegant, right? You read down, very human. But like, I was just thinking, reading this code and misreading the code, by the way, how much worse that idea is. I'm sure for like small Kamers, it's probably fine, but like, I wonder how bad it would be for a 9mer to do that. That's suboptimal because the vast majority of substrings you're searching for probably won't exist in the text. That's true, but I think Big O would allow me to know how much worse that is, right? Or is it the same order? That's what I'm kind of curious about. Like, is this a bad idea or a terrible idea? My solution is n times k to the power of 2. That's pretty bad. That's pretty bad, I'd say. Yeah, so paying attention to how algorithms work can that's the kind of optimization I think that programmers love. It is fun to think about, but quite complicated, I think. I think programmers are professional lazy people. I don't know if you agree, because they're always trying to find ways to cut corners. It might not be lazy to do that, but to write something that is the maximally laziest thing that can happen, I think is interesting. Professional, I don't even know what to say. Loungers. Okay.
Well, I must say it is pretty cool when you can get something to run faster and more efficiently, or if you also think about game optimization, when a game is not well optimized, at least in the algorithmic sense, then it runs slowly and lags. There's other ways it can be unoptimized, but that's neither here nor there. But yeah.
So I'm gonna start a poll in the chat, just to see where our mood is in terms of how to finish this stream. So we have two options. We can just continue to hang out and talk idly about bioinformatics or cholera. If you want to pitch more drug design ideas to me, that'd be kind of cool. But Resta also has more slides prepared about the profession, that might be worth going through if there are chatters in the audience that want to know more about like the practical side of bioinformatics or more about the lived life of one. Yeah, I think it's I just want to thank Resta as we're winding down. I mean, we're gonna hang out after, you know, the slides or whatever, but just wanting to thank Resta for enabling me to have this really fun look into how bioinformatics works, because I would have spilled spaghetti everywhere probably, because like if you hadn't done this, Resta, I might not have done a bioinformatics stream, because that's how uncomfortable I would have been with that idea. I'm interested in what life would have been like if I had actually found a career in bioinfo. Yeah, I don't really know. I don't know if the tech layoffs have affected biology as much as other computer programming based industries. I don't really have insight into that whole thing. I do know that when I was in graduate school, bioinformatics was like super hot. Like if you could get your hands on a bioinformaticist, you know, because we were all doing like big data kind of stuff, it was really enticing. And that's why I wanted to do bioinformatics for a little bit as an undergrad, because I was like, well, I like biology, but I also like getting paid. But then I found out I was terrible at it, so that stopped that in its tracks. I voted to see the slides as to see into the professional slide, but would love to just generally hang out and ask questions. Resta, how are you feeling, just like constitution-wise? Are you okay to...
I think I'd be good for some more voices doing okay.
Okay, great. So it looks like there are slightly more people that want to learn more about the profession, and I know you already did the prep for the slides anyway, so let's talk a little bit about what it means to do bioinformatics, and then, you know, if you have to go, then that's chill, but I'll stay and hang out with chat for a bit after those slides for sure. Okay, I'll end the poll there. So let's continue on and see what it's like to be a bioinformaticist.
Okay. Let me just do. Okay. So let's talk about bioinformatics as a field. What is bioinformatics? Well, one way we can talk about bioinformatics is the data that's used in bioinformatics. So to answer that question, we're back to the central dogma of molecular biology.
I love this slide.
Yeah, oh shit, here we go again.
Here we
back
go
to
again.
The central dogma. There's no escape from it.
No, it is very central.
So as you guys know, DNA is transcribed into RNA, and RNA is translated into protein, and then proteins do all sorts of things, such as produce metabolites. And if you've seen the previous stream on genetics, you'll know that there's many other pieces to the puzzle as well, including epigenetic modulation, post-transcriptional regulation, and other things. Research into each of these processes has, in turn, given rise to unique fields. For example, the study of all the DNA belonging to a particular cell, its genome, is called genomics, as you can see there. Likewise, there are fields for all a cell's epigenetic modifications, RNA transcripts, proteins, and metabolites, that is, epigenomics, transcriptomics, proteomics, and metabolomics. There's even more variations of this, as you may remember from early in the presentation when I mentioned metagenomics. Well, we can also have metatranscriptomics. There's also something called pangenomics. There's a lot of omics. But in any case, these fields produce a lot of data. And that's where bioinformatics comes in. From DNA and RNA and protein sequence data, which you can store in FASTQ format, as you can see there, to epigenetic modifications and transcription levels, which you can view in genome browsers, to mass spectrometry and nuclear magnetic resonance data of proteins and metabolites. There's just a lot of data and a lot of different kinds of data. Of these, however, I would like to highlight sequence data. But before we jump to the next slide about that, let me get you guys thinking. What are some cool applications that these kinds of data might be used for?
I love this question, just because when I was in grad school, everybody wanted to do omics work. In fact, I think that the power of omics work was so appealing that it actually caused some of us to get a little intellectually lazy, because it was pitched to us in graduate school that dude, omics is gonna solve everything. If we just learn everything, then we're gonna be freaking good. So I love this idea of understanding the power of wielding data in this fashion. So I think it's really neat. I also started a poll, sorry to do it in the middle of you talking, Resta, but all of a sudden I just felt very conscious that I hadn't polled the chat in terms of their background. So I was very curious as to see what the split is between the backgrounds in chat, but people have made it to the end-ish of the presentation, to see whether or not we're talking mostly to biologists or to computational people. It seems like there's a pretty decent spread of people who have came in maybe from the biology side to learn more about programming from the biology side, et cetera, and a good chunk that have neither background, which is also very interesting to know. Hopefully we've sold you at least one of these. Morju says protein dynamics / folding is a part of bioinformatics too. It's a big part of drug design now. Yeah, AlphaFold has been really, really pivotal in a lot of hypothesis generating applications. I used AlphaFold quite extensively in my prion video because there doesn't exist a full-length prion crystal structure, because you can only visualize a part of a protein that is not floppy to the wind, right? And AlphaFold lets you mathematically guess at what it looks like in reality, whereas crystal structures are very accurate, but you have to make some sacrifices. You have to make it crystal, which isn't a biology environment, right? Cryo-EM, you have to do it really cold, and that's not really the same as biology. AlphaFold lets you do it with math, which also isn't really biology, but it lets you start. It's pretty powerful in that regard. They got a prion EM of prion disease fibril. That is true, but again, since the conditions that are required to do cryo-EM aren't exactly because you can imagine, to do cryo-EM, you need to go way cold. You need to go, I don't remember. It's somewhere in the minus Celsius, absolute zero, not quite absolute zero, but very, 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 very cold to get those proteins to work. I'm not a cryo-EM expert, so I can't say beyond. You need to get it real cold. And as you know, inside the cell, it's not really that cold in that environment. So it's a very good way of analyzing a protein's native structure, but it's not one-to-one. And again, that's why these complementary approaches are so valuable, because if you do a whole bunch of science and all of that science starts to agree with one another, then you could start thinking, oh, maybe I've stumbled into the truth here. I think it's cool how omics led the way to discovering new bacterial defense systems, drug resistance, anti-phage, which is cool in itself, but we can use this knowledge to target those systems. Yeah, omics stuff is nutty. Yeah, genealogy and stuff too. Yeah, that's where my brain first went to, is DNA is data. And I love the slide too, Resta, because you have pointed out that there are so many different omic fields. So if you like thinking about big numbers, big data, there's a fertile ground of research in understanding data at this scale for biology.
Mm-hmm.
Yeah, anything you wanted to add, Resta, or any answers that you wanted to highlight from the chat?
Speaking of AlphaFold, I did really like that picture that Clockwork modeled using it in the protein stream. That was very cool.
Yeah, Clockwork's great. If I he's so nice. He offered to help me with protein modeling if I ever wanted to do that in my content, and I was like, thank you. I think learning from YouTube tutorials is really hard, and having someone that I can ask to just show me how to do stuff is like really kind, because I am like a computer dummy. But yeah, shout out to Clockwork Show, producer of, I think, the best video on ATP synthase on YouTube right now. Very fun.
But yeah, in addition to all the answers that chat has provided, I want to share some of my own favorites. For example, extreme microbiology. So I mentioned earlier that majority of microbes are uncultured. We don't have much information about them, both genomically and physiologically. And so bioinformatics is very relevant for studying the extremes of life. For example, at the bottom of the ocean, you have the seafloor, right? Well, like far, far below that, you have what's called the subsea floor. And you have microbes there that live so long and have such slow metabolisms that they're technically alive, but barely. And then, of course, you have microbes that, like other extreme conditions, such as high temperatures, those would be thermophiles. High salt concentration, those would be halophiles. Some microbes like really cold temperatures. And there's also research into microbes that are growing on the International Space Station as well, being constantly bombarded by radiation. So, and that in turn kind of branches into astrobiology. So that is one of my favorites. Another would be paleobiology. So I mentioned earlier that there's bring mammoths back, or these elephant mammoth hybrids. And and also, in addition to the mammoth stuff, we can also uncover ancient human and animal migration patterns. Figure out there where you can see the dispersion of Africa and going to Europe, going to Asia. The genomes, like you get that information, you process it in different ways that I'm not super familiar with, but after doing that, you can then remap these like huge events that changed human history. Those brave people were like, I'm getting out of Africa, and became Europeans and Asians and Native Americans.
Yeah, that's really cool. I always find like every chance to plug like spiritual connection to biology is that like there's a bit in my upcoming video about the immune system in the gut and how the immune system sort of identifies which bacteria are good and which bacteria are bad. And I have this like long-winded speech in the middle of it just being like, I don't know, physicists don't have a monopoly on like finding spirituality within their study, right? Because I think that using genealogy and stuff like that to track the movement of our ancestors is really a way for us to connect to our own evolutionary history. And I think, I'm excited to see sort of how that plays out. Just because, it's sort of easy to forget that we are products of surviving. By virtue of existing today, we are lineages of the most extreme survivors. And so using science to sort of uncover how that story happened, I think is always going to be very fascinating.
Mm-hmm. Another thing of paleobiology as well is that if you're of European or Asian descent, it's been found that you have a certain percentage of Neanderthal DNA in your genome, which is pretty cool.
Yeah.
Anywho, yeah, more juice, forensics. So bioinformatics is also applicable to solving crime, such as identifying cold case killers and the like. Um, it's also been applied to personalized medicine. There's been a lot of promises made that haven't quite panned out, but progress is being made. But the general theme there is to provide patients with more precise, potentially life-saving treatments. And then as a last example, one that's actually very applicable to the presentation that we had today is wastewater monitoring. For example, you can monitor cholera in wastewater, and you can also monitor COVID. In fact, I went to a VTuber Expo in San Francisco not too long ago, and I contracted COVID from it, which was no surprise because looking at the wastewater data, COVID levels in San Francisco had reached like the highest levels ever since the start of the pandemic, if I recall correctly. And that's that in turn delayed this particular collab, which was supposed to be last week, but um, but yes.
The city by city, state by state wastewater databases are public actually. So if you just search wherever you live wastewater COVID, you'll find the wastewater monitoring. I actually saw that when I got COVID, like a day earlier, or not even maybe like 3 days earlier, I just had the urge to check what wastewater was like in my local area, and it was on the rise. And then I was like, oh god, it's coming. And then not like 3 days later, your boy got the vid. So wastewater, if you are like planning a trip or something and you want to remain COVID free and you want to like have an informed understanding of what your chances of contracting COVID in the public are, go look at the wastewater. It's a very good indicator of how much of it is out there. And it, at least in our cases, Resta and mine's cases, we saw it coming.
Yep.
Awesome.
But yeah, okay. Moving on. Where's my cursor? Okay. So the reason why I wanted to highlight sequence data specifically is that starting in the early 2000s, there's a huge surge in sequencing data driven by a huge increase in sequencing affordability. To give you a sense of how incredibly fast sequencing technology developed, let me walk you through this graph. So there are about 3 billion base pairs in the human genome, and the first draft assembly of it costed the International Human Genome Sequencing Consortium about $300 million US, which by the way, you can download these specific genomes, these early assemblies on the internet for free, which is a very pricey file. In comparison, though, the current price of a genome hovers around $1,000, which, to give you a scale of this, as first said by Dr. Yuan Ashley, if the automobile industry progressed at the same rate as genomics did, a Ferrari would cost about 40 cents today.
Jesus.
And how is this possible? Well, through the invention of next-generation sequencing platforms. What are those? Next-generation sequencing, or NGS, these days is monopolized by a company called Illumina. There actually used to be another company called Roche, and they had their 454 pyro sequencing platforms, but they don't really used all that much anymore. Anywho, over here is the Illumina HiSeq. It's a machine about the size of a mini fridge, and it has a flow cell, which you can see here, through which DNA is moved with microfluidics. One of these fellas can generate up to 600 gigabases, that is 600 billion bases per run, using a single one of these flow cells. As a certain meme goes, that's a lot of data. And it's through this huge amount of data generated by machines like this that the field of bioinformatics really has come into the limelight, because labs want to sequence DNA and they need people who are able to work with these huge amounts of data. But yeah, any chat? Yeah. After this, let's see. Cursor. There are some other aspects of the field of bioinformatics that I'd like to touch on as well. For instance, in today's presentation, we found a way to find DNA boxes within a bacterial genome. Well, to do that, we relied on a program like OraFinder to determine what is the ORC region within Vibrio cholera. Before that, the genome that had to be fed into that program needed to be quality controlled. For example, when you use one of those Illumina machines, there's a certain pattern in the data that the longer the read that you sequence, that is the fragment of DNA that you sequence, the base calls at the tail end of that will be less accurate. It's through a process called out-of-phase accumulation. I should have put a picture of that here. But in any case, those are some things that would have to be done before we run our DNA box finding algorithm. But then after that, we'd probably want to turn that data into a nice table, especially if, let's say, we're looking at like hundreds of different strains of Vibrio cholera or something like that, or different bacteria in general. You want a way to tabulate that, and you'd probably also want a way to visualize that. That's where the whole field of data visualization comes in. Now, this workflow is pretty linear, but as you can see here in this example pipeline, the different steps when you do bioinformatics can get very complicated. Different steps will rely on previous steps, and it can become quite the web very quickly. However, being able to construct one of these using either Nextflow or SnakeMake is one of the key bioinformatics skills in industry that For example, if you apply for a bioinformatics job, most of the time they'll be like, oh, do you have Nextflow experience? Since that's kind of the gold standard. Snake and Make is also pretty good. It's great if you know Python already, but people don't take you as seriously unless you use Nextflow. In addition, scientific reproducibility is also a very important facet of bioinformatics. For example, sometimes people will make a program and publish it and post it on their lab website. Well, what happens when that lab website changes URLs or goes down for some reason? Well, then the world no longer has access to that particular program. So it's important to upload things to upload your bioinformatic programs to repositories such as GitHub that will actually stick around. And I really mean stick around because like sometime back, all of the code in GitHub was actually recorded on like a bunch of hard drives or something, and then put inside of a frozen mountain in Northern Europe or something. I forget the exact details, but it is literally in cold storage.
That's insane. I had no idea that they shoved all of the data and it's that's nuts. I love that. Oh wait, sorry, something happened to the presentation. What did I do? What did I do? Okay, it's back.
Yeah, kind of like the seed bank to me. Also kept very, very cold.
Yeah, I love this idea of open of open source science. Like this attitude that people who work with code have of sharing their code is really encouraging, especially because I have stories of working as a scientist in a lab in graduate school of certain labs having certain reputations being very stingy with sharing stuff, like for example a specialized mouse line, a mouse with a specific genetic mutation that would be perfect for my project, but their lab having a reputation of never sharing that mouse just really irked me. I was like, I think it's really encouraging that there's this culture of open and sharing, as there should be in science, right? So I'm going to be a little salty about not getting my hands on that mouse due to just, that lab feeling insecure about their ability to publish. But, you can't blame them because I talk a big game about like academia is great. Learning is great. Being a student is great. Like learning and pushing yourself and really getting deep into a field, that part is great. The actual drama behind academic life is maybe not for everybody because sometimes you run into people who won't share their bias, stuff like that. Exactly, share your bias. All right.
Another reason why it's important to make your software open source is because in terms of software, a lot of bioinformatics programs are kind of like the Wild West in the sense that they aren't often held or programmed to standards in modern computer science industries. And so, and furthermore, once someone publishes, at least within academia, they don't have a whole lot of incentive to actually maintain that piece of software over time. But if you post it to GitHub, then it's open source. Some people might take up the project and like there might be a new maintainer, for example. But yeah, just in general, both for programs and data, it's important that they follow the FAIR principles, which means they're findable, accessible, interoperable, and reusable. And that's another standard that's that to aspire to, at least.
I think I also like the description of bioinformatics kind of being the wild, wild west, because some people really thrive in that kind of environment, right? Like, in terms of VTubing, educational VTubing right now in the indie space is sort of the wild west. And I love this feeling of like, we're seeing more and more educational VTubers every single day, but we're not really like an established niche on YouTube yet. There aren't rules. There's no established culture. We're kind of all figuring it out at around the same time, which is very fun because you can tame the wildness in a way. The more you work, I don't know, that's a weird way of putting it, but this idea that you are in an open environment where you have the ability to flex your creativity to solve problems that need solving, I think is appealing about that field.
I'm certainly excited to see how things will progress over time, especially with V theorem too.
Thank you so much. There may or may not be news in the future, in the near future, about V theorem, but it's certainly an exciting time, I think, to get into educational VTubing. Be very neat. All right. Let's see. Anything else down here? Oh, that's it. It looks like.
Yeah.
That's awesome. Thank you so much, Resta. Yeah.
Well, thank you for having me.
A little bit of behind the scenes, Resta. This was like your. I know you had experience with education before, but is this like your first, like public education kind of thing?
Technically, yes. There were a series of lecture-y things I did that were uploaded to YouTube. But at the time, I didn't really like know it would be uploaded. They just kind of told me after the fact. So this right here is certainly a very unique experience and has challenged me in a lot of new ways.
Yeah, this was the first one that you consented to. I didn't coerce you to. Ways to find Resta content if applicable. Thank you so much. Let's go ahead and plug your channel, Resta, if you want to. I think it's in the stream description as well.
Yep. In the doobly-doo.
In the doobly-doo, correct. Yeah. I think that. First of all, chat, please give Resta a round of applause. We're not in a conference hall, but I certainly have been championing for Resta because, again, it's an intimidating space to step into. If you just think about like the fact that she willingly took on this idea of leading this stream, because usually I do most of the work, and then so I can like structure it and stuff like that. But and she, I wish you could know how much behind the scenes work she did, because it truly is. I'm very lucky and happy to have you as a collab partner, just because she took into things like consideration about like how I interact with chat and tried to model it after sort of the style that I like to do, which is like way more work than I could ever ask anybody to do. So I'm very happy to have that level of consideration when it came to putting this together. I think you did a great job, Resta. Yeah, Resta quizzed you too. Yeah, exactly. Like, I think those are the fun parts of the stream, though, like thinking of ways to make the story of science compelling, just so that it doesn't look like your lecture too much, because the educational we call them pedagogical goals, I think, of lecture in a university versus here are slightly different. I say it every stream, but it's important that people don't get it twisted, that we are here to keep you engaged with science, and if you learn something, that's extra credit on top, whereas in class their goal is to make sure you learn something, and if you keep engaged with it, then that's extra credit, right? Like, you are there to pay for something that turns into a degree, which turns into a job, and I think that if you go with both, then I think there's a happy medium, right? You can learn new things and still have fun, and I think it has been fun to sort of revisit. Like when Kamer showed up in Resta's practice stream, I was like, oh my god, Kamers! I just like had like flashbacks. And like, oh, I actually found those were cool at some point before having dropped bioinformatics. So it's been super fun because we're revisiting these ideas. At this rate, I might end up marrying science. Not if I marry science first. Hopefully Toxo did not hear that. Oh no, science NTR. Yeah. So I think we'll take other questions about cholera, about bioinformatics, or we just stay and chat about biology or anything really. And until Resta, you have to go, or you know, if you want to stay and chat, that's cool too. Oh no, she's here.
I also just want to add, thank you so much for hosting me, Phi. Like,
yeah, no problem.
As I mentioned, this was challenging to me in a lot of new ways. If you guys see my Twitch, you'll see I'm a bit of a more traditional VTuber in terms of gaming, in terms of like personality and all that. But there's been this science part of me that it was always very intimidating, like how to put that part of myself out there. And I've always kind of wanted to, because while I was pursuing my degrees, I was a big fan of science content creators like The Brain Scoop with Emily Grassley, Veritasium, Kazert, and Alex Danis, who is one of my faves, and who I also got to meet in person once. That was super cool. But in any case, when Phi reached out to me for a collab, I knew that this was my shot, and I wanted to do right by it, and here I am. Yeah.
Yeah, that's awesome. Of course, I do whatever I can to encourage more VTubers to at least enter this kind of space. Before I ask you a follow-up question on something that you said, just because I think it's kind of interesting, Mute did have a question that we couldn't get to before, but I certainly have an answer for this, but Mute's asking if you've interacted with machine learning in any kind of way. I actually have in my line of work, but have you done any kind of machine learning?
Depends how you look at it.
Ooh, biology is a spectrum. Let's go.
In my bioinformatics algorithms class, and if you actually look at the textbook, you'll see that there's a section further deep in the textbook. I think it might even be in part 2 of the textbook, but it covers using hidden Markov models to, again, find hidden messages within DNA. In fact, I should not say that because it'll dox me, but what I will say is hidden Markov models were very important to the Human Genome Project. And that's as much as I'll say. And so that section is kind of like pre-machine learning, like building it from scratch. But in terms of like actual machine learning libraries, I haven't really worked with those in any significant capacity.
I'm wondering if I can find an example. I can. Thank you, Wikimedia Commons. We love things that I can show on screen that won't get my whole thing demonetized. I actually have worked a little bit with machine learning to no avail, which is why I'm a little... Scorned is not like the right word. I'm a little skeptical now because when machine learning was sold to me, I was like, oh, I'll just do machine learning and my problems will be solved. Turns out it's not that straightforward. My work was in neural immunology. So for a long time, we thought the brain was immune privileged. Turns out that's not the case. Turns out that the brain actually does have an immune system, and that it does interesting different things that you wouldn't really expect the immune system to do. And so part of my research was understanding the synapse and how the immune system interacts with the synapse. So if I can show this picture, this is a neuron. And so I'm sorry if you're red-green colorblind, because again, biologists are very not friendly to red-green colorblind people, but you can see that there are these red dots. We call them puncta. And so I was using a machine learning algorithm to try to identify how big and how many these dots are. Each individual one of these is representative of a point of connection that this neuron can make with a different neuron, this so-called postsynaptic density. This is where a neuron would hook up to another neuron. And I was interested in the number and amount of these little connections, but it didn't really work, so that was a little bit of a bummer. Biologists aren't great at accessibility. Yeah, in my class I always say to my students to use blue, orange, or something better than red, green, because that doesn't really work anymore. We should try to be doing a little better with regards to making sure that everybody can see the pretty pictures that we all get to enjoy. So it was a good application for that sort of problem, but it didn't end up working. I don't know why, but I just decided to use more traditional tools for that kind of thing. Resta, I had a question for you with regards to you saying that there were challenges, and it might be helpful for the audience to hear what those challenges were, not to put you on the spot and be like, you struggled, let's see. But I think it makes for genuinely good content to sort of walk through what made it challenging, to sort of appreciate how you may have overcome some of those challenges.
Okay.
You don't have to answer
It's a
if you're
bit
uncomfortable with
personal,
that.
but one of the things I can say is academia hasn't been all too welcoming of my interests in like anime and and Japanese culture and all that. Like I remember this one genetics class where I just mentioned the word anime and I got the stink eye like, oh, who is that loser? Um, and so I've because of these experiences, I've kind of separated these two parts of myself a lot mentally. If you watch my content on Twitch, you'll notice I have I tend to speak like in a higher pitched voice than I did during this presentation. I tried to do that here, but my the way my brain has set this up over time has internalized this is just too strong, and I wasn't able to fight it. But like yeah, and the reason why I used a lower pitch voice here is because like that's the voice that I use when I'm talking with my colleagues, and because if I use a higher pitch voice, then I sound like an anime person, and it's harder to take someone seriously. Like that's I mean, I wouldn't judge someone who did, but I know other people would, and among other very sussy things I've heard in academia. But yeah, bringing these two parts of myself together in this form, it has been a big step for me personally. And I just want to thank you again, Vi, for helping make that possible.
Oh, absolutely no worries. I think it's really cool actually to see this part of you because, one of the things that I had pitched in terms of coming up with ideas for this collab was like, you've done work or you've had experience with plumbing the depths of the ocean using bioinformatics, as you had said. That kind of niche expertise and like shows, I think, that you are very capable of handling yourself in this sphere. Just like, yeah, want to reemphasize how impressed I was while talking to you and planning for this collab, not only due to the depth of your knowledge, but also the hunger, I think, to be able to put this together. Nothing but respect. Very, very cool. Yeah, that's really, and of course I'm sorry that you experienced that with academia. I also felt like I had to mask quite a lot in terms of my interests as a graduate student. Some part of me will never be able to completely mask who I am. I just can't do that. I'm physically incapable of doing that. But I was very lucky to be in an environment that was like very accepting of the weirdness. I think I'd mentioned on stream actually that I had like a breakdown during graduate school interviews. I just like went to the bathroom and just had to like cope on Reddit because everybody around me seemed so normal. I don't know if I told that story on stream actually. Everyone around me during interviews, we were all, when you're at an interview for graduate school, you're there because they already want you. They just have to make sure you're not a weirdo at that point, which is how graduates like interviews go. That's not a secret. If they give you an interview, they already think you're qualified. You just have to not raise any red flags. And so being at the university that I went to, it was really tough to like function as a normal person, hiding parts of me that I think might have not been appealing. Being able to be in an environment where you can be that kind of true to yourself is like very, very relieving. I tell the story to all of my graduate student friends who I ended up going to the same university as me, and they find it very funny. But also they say that makes a lot of sense because I'm like the most asocial guy ever. I know it's a meme at this point that all VTubers are like, introverted and antisocial, but it's just being in that environment where everybody's trying to impress everybody else. Not only is that like kind of toxic, it also just had it felt bad in that case. Academia can be very toxic. Yeah, it can be, especially when you're trying to, I call it peacocking, like when you're peacocking for your professors, you're peacocking to other students. I just wanted to chat about, I just wanted to have a normal human conversation that entire weekend. The entire weekend, I just wanted to talk to another human being, but instead it had to be, oh, I published here as an undergrad. I'm like, God damn, that's impressive. I'd never published as an undergrad. And oh, I did this groundbreaking research in this cancer lab as an undergrad. I'm like, okay, I know I'm like trash. Like, shut up. I just want to talk about anime. Like, I truly relate to that. Just feeling of like, I just want to skip the part where we have to like figure each other out, which is why I have anime, like I have HoloLive figurines in my office, because I'm at a point where I don't have to. I'm blessed to be in an environment where I'm not going to be punished by anybody for having HoloLive figurines in my office, and I'm very lucky to be in that position. I hate interviews. Yeah, interviews suck. In a way, educational VTubing is a way to reconcile the professional part of you and the weird part of you. Yeah, exactly. Like, I think that's exactly right. Like, even if I weren't a VTuber, even if Toxo weren't so talented and lovely to rig and draw Phi, I would figure out a way to do this somehow, although it would not have been in this space, which, again, I'm very thankful to be in this universe where I get to do this and, make $1 or $2 doing it. Could not ask for... And, I'll save more of the sappy recap stuff for the stream I'm playing next week, which, of course, is the 1-year anniversary stream of making content on this platform. But it just reemphasizes that, like, this is the wild, wild west. It's such an exciting place to be. And, for all sorts of different reasons, there's gold in here, and I can feel it. The creative parts, I think, are really... important, I think, to just expression of who you are as a person, right? Like, I think there are for me personally, challenges in being a more traditional VTuber. I don't think I could have this audience looking or being any different. And I think that, you know, when I was brainstorming with Toxo about how this channel would look like, we kind of both agreed that who I am isn't an anime boy. It's like, and there's a lot of angst, I think, behind the scenes about like, oh, if only I were an anime boy, so and so marketing would be easier. But just accepting that, like, I'm not an anime boy. I don't think that that's appropriate. Other doctors in biology, is that a figurine of an anime girl shark? Yeah, my office mate moved in like yesterday, and he's got all these pictures of his lovely family and like biology merch and very impressive textbooks and, you know, philosophy books. Like, he appears to be a very smart person. He went to a very impressive Ivy League school, so he's got like an Ivy League lanyard like hung up too. And then on my side I have Fubuki and Subaru, and I think I have acrylics of Ame and Crony just like hanging out on my shelf. And I'm like, yeah, we have the same job, actually, is that you can be that and you can be me and earn the, you know, maybe not earn the same amount, but have the same title description. And very happy to be in that kind of space. You can be an anime man instead. Yeah. I have crude MS Paint level drawings of those kind of design sketches lying somewhere. Yeah. Oh, I have a story for you, Resta. As the pillow VTuber, I actually ordered a new pillow that is arriving today because I was diagnosed with mild sleep apnea. Like, it's very fitting to be collaborating with a pillow VTuber on the day that I get my fancy anti-sleep apnea pillow. Just to just abruptly shift gears from this like very tender conversation about acceptance to be like, I like snore a lot and I don't breathe when I sleep. So the pillows actually help incline. It helps incline your body in a way where you don't angle your chin downwards, which can happen if you're on a completely flat surface. And if you angle your chin downwards, you can close your throat. And if you close your throat, it can interrupt your breathing and cause you to snore. So there is that aspect. My doctor also gave me all of my sleep data, which is really fascinating. I love getting like biometric data on myself. Although I would never get my genome sequenced. That's something. Would you ever get your genome sequenced, Resta?
I'd have to read the terms and conditions for that, since I know lots of those services are the type of services where you are the product.
Yeah, exactly, right. Like that's also my fear, is that like, I don't know, there could be one day in the future that 23andMe takes your data. I'm ruining my 23andMe sponsorship right now, but I don't care because I still believe that this is true, unless they somehow convince me that it's not true. But if an insurance company ever got a hold of your DNA, that would freak me out to no avail, right? If a health insurance company understood what your DNA looked like, that would I would not feel comfortable with that happening. 5 genome leak, exactly. I don't want my genome to be leaked. That is true. Too much data can lead to paralysis. Careful. Yeah, so there's this thing called analysis paralysis that I was experiencing a lot as a graduate student, where there was okay, so this is peek behind the scenes when it comes to a PhD. If any of you want to think about doing that, is you have to come up with a new idea. No matter what PhD you do, the idea that you have to do is new. It has to be new. It cannot be redundant. You have to read everything in your field to make sure that it is in fact new. And when you get down to honing in on the project that you pitch to your committee, it has to be good and it has to be clear. You cannot pitch everything in existence. You have to narrow it down to something. And having analysis paralysis of like, I have so many different directions that I could go in, and narrowing it down to just one plan and a backup plan is overwhelming. I experienced a lot when I was thinking about doing psychology as a profession too. So there's an alternate universe where I could join Sidearray as another psychologist VTuber, but that ended up not happening. And I think it's a lot easier to get taken, like, to get football tackled into a place you don't want to necessarily be in psychology, just because it is so sticky and messy, I think. I don't want Mark
Oh,
Zuckerberg
hi.
knowing my DNA sequence. Yeah, me neither. What's up, Resta?
Oh, Morgie's asked why I chose to be a pillow VTuber, and I do have a lore snippet I could read.
Ooh, that's fun.
Okay, but anywho, can people put links in chat? I'm gonna try. I know on Twitch I have links disabled, but
I think we're okay on YouTube, although I'm not 100% sure.
in any case, this is the link to my VTuber page, and you can see down in the lore there, it has a bit about my in-lore background. And I'll read through it. Can a pillow ever gain sentience? Well, common sense would dictate no. Yet ever since the VTuber uprising dawned upon our world, we've seen a drastic increase in phenomena that defy our previous understanding. For instance, phenomena that could turn a pillow into a person. And that's what happened to Resta Makurabe. Once just a pillow, now a researcher of a dreamscape that might hold clues to the origin of the VTuber uprising, and in turn, herself. Such research is unprecedented, so she's turned to live streaming to help document life in the dreamscape. Perhaps one day Resta will uncover a breakthrough, or perhaps you, fellow dreamscaper, will discover something even more special along the way. Signed, Cosmo the Axolotl.
Oh, that's what the axolotl is? That's nuts. I actually didn't know that about your lore. That's awesome. I thought it was just your buddy. That's awesome. I love that.
Also, the symbols on my sleep mask and at my side are from a flower associated with dreams called the white egret orchid. It looks like a bird. Very cool orchid.
Yeah, I have to say that, like, one of, I think, the most impressive aspects of you as a VTuber, Resta, is your incredible visual brand identity. Like, if you visit Resta's stream, it's very expertly done. It's very artistic, and honestly, like, my brain can't work that way. I'm still pulling up stock photo images from from Adobe because I don't know what I should put in anywhere. I think Resta's visual IQ is like super high. Yeah, that's amazing. I love that.
Thank you.
So Mandrake asked what I roughly did my PhD on. It was I'm not really doxable, so I could go into a little more about it, but it's essentially asking the question, what the heck are immune proteins doing in the brain? Immune proteins, of course, they help us fight bacteria and stuff, but there are no bacteria in the brain. Probably no bacteria in the brain, not as far as we know. But we do know that the brain makes immune proteins. And so the question of my PhD is asking, what the heck do those immune proteins do if they're not fighting viruses, if they're not fighting fungi, if they're not fighting protists or amoeba? They actually do brain things. They help you with things like trimming synapses to help coordinate your neurons in terms of their development, and a whole bunch of other things like recovery from brain injury as well as worsening brain injury depending on the context. I just found that appealing, right? Because I had a choice between a couple of different labs. And to me, the unexpected nature of the immune system having different, like, nighttime roles in the nervous system was so interesting, right? This idea that these proteins that we know and love from immunology are doing wildly different things in the brain is nuts. A microbiome in the brain. There's a virome of the brain for sure, and I think there was a T-cell member that had suggested that I look at the bacteriome of the brain, and that was something that I had never heard of before. So one day, as a member's suggested stream, which of course everybody will be able to see in the public, we'll ask the question if there are bacteria in the brain. It'd be pretty interesting. Meningitis is in fact viruses or bacteria or fungi. I think there can be either of those pathological agents causing infection in the brain. That is correct. But they're there when they're not supposed to be. The question of whether or not there are bacteria in the healthy individual in the brain is a lot more of an interesting question. No bacteria. I would like to know more about the no bacteria in the brain. Yeah, as Morju said, there's the blood-brain barrier. So that is a physical wall that prevents stuff from the circulation from getting into the brain. It also prevents the immune system from getting into the brain. Right? It's just a physical wall that is impermeable to a lot of things besides the nutrients that the brain needs. Once that blood-brain barrier is breached, and you get like, for example, neutrophil. I think for the brain it's mostly T cells, but if you get infiltration by the by the immune system, you actually have a lot of problems. So I think, and I could be changing the tone when I actually read the bacteriome paper because I just skimmed it, is that they find that there is bacterial DNA in the brain, and so maybe there actually is a bacteriome in the brain, and we haven't found it because, and this overlaps with Resta's interest, maybe those bacteria are unculturable. Or it could be a lot of hooey. You could just find bacterial DNA because bacterial DNA circulated to the brain, and that would be less interesting. And figuring out which one of those truths is real, I think is going to be very interesting. I did mention the brain
Another
microbiome that time.
possibility
Yeah, that's
that
what.
is
Sorry.
oh, another possibility that is actually a big problem in the field of metagenomics is contamination. Either subjects like human researchers contaminating their samples, cross-contamination between samples, contamination from reagents that are used like to process the samples. And then there's the whole bioinformatics side thing too, where you do got to be careful.
Yeah, that is true. I always assume that the scientists did an okay job, like physically, because there's only so much that I think you can be critical of in a paper, unless you are reading it for your job. Like when I read for Phi the Neutrophil, I don't treat it the same when I read for my job. I'm way more careful for my job than I am with Phi the Neutrophil, right? Because asking whether or not a paper that is published had a potential contamination issue is not something that I would worry about for public content. But for my job, it's really, really important to practice what you preach. Because I just kind of give the scientists leeway, right? Like, hopefully you did the thing correctly and it's fine. But when I teach students, it's like, you have to think about everything, whether you want to or not. Otherwise, you could spend several years of your PhD tackling a hypothesis from a sketchy paper whose methods were not super reproducible. Not like I'm speaking from any sort of experience or anything. Ha 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 ha. There's an interesting pointed question. I don't know if you know the answer to this because I think this is actually in between our expertises, Resta. What techniques do bioinformaticians use to keep track of euchromatin and heterochromatin patterns and their histone modifications? I don't know exactly from my end, because the only thing that I'm aware of with chromatin stuff is stuff like ChIP-seq, which I think answers.
I've heard of Hi-C.
Hi-C, do you want to speak a little bit about that if you can?
It is very cool. It gives you information about the physical, like, what's the word? The actual physical locations of where certain DNA fragments are. For example, there was a marine microbe that I was interested in learning about that is known to have a symbiote inside of it that is very important for nitrogen cycling in the ocean. As you may know, nitrogen, the conversion of nitrogen from abiotic forms to biologically accessible forms is very important, not just in the ocean, but it's also like with the whole Haber-Bosch process to create fertilizer for the Industrial Revolution and all that. And so one of the approaches that was being looked into, not by me, but by other people, was trying to use Hi-C or a variant of it like 3C or meta 3C to disentangle the genomes of the host cell and the symbiote of interest. Yeah, that's what I can say about that.
In terms of understanding the function of chromatin, I believe, and again, this is why I said it in the genetics stream that my genetics is actually quite weak, but I believe that in terms of sites of transcription, and someone in chat can correct me if I'm wrong, when you have unwinding, so chromatin are these proteins that your DNA is wound around, and when local areas of your chromosome are unwound, that's when proteins and stuff can get access to your DNA to start making things. So they are looped around histones, and when they are looped tightly, that DNA is inaccessible. So you want to unloop in certain areas. I believe that is correct.
There's a kind of sequencing that allows people to study that too, called ATAC-seq.
Let's see.
ATAC.
ATAC. Interesting.
Yep. Assay for transposase accessible chromatin sequencing.
Oh, nice. That makes sense. It's also cool, I think, to talk to a colleague. I know that a mutual streamer that we're aware of, Spolax, who does microbiology work. It's just nice to talk to other biologists, especially when we're not stressed about our work as biologists, because like I don't think you should do science I don't think you should do biology specifically if you want to make money. I think you should only do biology if you like biology, and not being pressured in an environment where like you have to, you know, do scientific work, but just kind of geek out about science stuff. And like I don't know, can I talk a little bit about something that I chatted with you a little bit about, Resta, a potential collaboration, or do we want to still keep that a secret?
Given that I'll be pretty busy in the months ahead, let's keep that one in the bag for now.
Okay. So we are not super sure whether or not a collab will happen in the future, but to summarize the thought is it's nice to talk about stuff that you like with people who like the same stuff, and it's especially nice when we are coming at it from slightly different angles, right? Because you know a lot that I don't, I know a lot that you don't, and it's just neat because we're talking about the same thing, but there's no one person that can know everything, and it's very neat. Looks like Mute has a question. Why would there be a difference between public-facing studies versus academic science-facing studies as far as possible study biases? Shouldn't be both just susceptible to problematic actions? The thing about the publishing system is you can record data, and the saying in science is data don't lie, right? Data doesn't lie. People do. Not on purpose, not all the time. But if the data will reflect the exact conditions that were done. And some scientists are better than others at keeping track of exactly what was done and how careful it was done. So you still get things like contamination that can happen, especially if there are no controls for testing for that kind of contamination involved. So, obviously you don't want to publish a paper that has contamination in the results, but that doesn't mean it never happens. It's not always the easiest to catch, especially when there are things like mycoplasm that are really hard to catch. So mycoplasm are tiny, barely living organisms that have a minute genome and a very tiny body that you can barely see under a microscope. And you need special kits to detect that level of contamination. And I didn't know about them before because canonically, the way I learned about contamination was, when you're growing cells and the phenol red is orange, indicating a very acidic environment, it means you have bacterial contamination or fungal contamination of some sort. But mycoplasm, you actually have to test in a completely different way. So there's also just like skill issue.
The whole data doesn't lie, people do reminds me of some controversial papers where the authors were discovered to have photoshopped some gels.
I guess the real data don't lie. The real data that is the true data does not lie. And I think people say that because when stuff, I almost said a bad word, when experiments aren't working out, it's very tempting to blame the data for being bad and then try everything exactly the same way and hope that something changes. That's a thought pattern that early career scientists can make. So if I do it again, it'll be different. In my class, if a student so we asked our students like, okay, this experiment didn't work out, what would you do differently? And without fail, every single time there are a couple students who go, oh, we just do it again because it's low end, maybe it's variance. And I'm like, the data don't lie. If you do it exactly the same way, the same thing is going to happen if it didn't work the first time. So you have to think critically about how to set up your stuff where A, the data isn't lying and you aren't lying, but B, the data shows something useful. I'm especially... Oh, so Morjuice has a question, I think, for you, Resta. Is it coincident that ATAC also uses nucleotide letters?
You'd have to ask the person who coins that word.
That is fair. That is fair. Marlon asks, I'm especially curious about the patterns of protein production in microglial cells in the brain, but that might be too big of an ask. So the question is a little imprecise. What do you mean by patterns of protein production? Because that's kind of vague. I did work quite a bit with glia, microglia. So for those of you who don't know, even though the brain is immune privileged, or was described to be as immune privileged because you could not detect immune proteins in the brain until recently, in terms of improved techniques. Okay, let me back up. For a long time, people thought the immune system wasn't working in the brain. I already said that. But even before that, we knew that the brain kind of had its own immune system. In fact, the brain has its own immune cell. It's called the microglia. And what this cell does is it trolls around the brain, it patrols the brain, and it eats debris and stuff. It is the equivalent of something like a neutrophil, but for the brain. Maybe one day we'll see a microglia, little phi guy. And so I am somewhat familiar with microglia, so I'm gonna have to... I'm mostly curious about the role of microglial cells in adjusting neuron connections, since I understand that's one of the roles of the microglia cell. I love that question. So I believe I teased this in the protein stream with more juice, with clockwork, so the immune system, right? So the immune system has these proteins, and there are these proteins that make up the, I forget what it's called, the attack complex. And it's a bunch of little protein fragments that activate a bunch of different pathways that ends up constructing this Gundam that punches holes through the bacteria. And so this attack complex is useful for blowing bacteria up, but also in neurons will actually make components of this attack Gundam on their synapses, which actually binds to a receptor on the microglia. And what is, I believe, the latest hypothesis or model as to how this works, is that the microglia sniff around neuronal synapses. They just kind of crawl along and they sniff the neurons. And if they find one of these Gundam pieces on the synapse, the microglia will actually eat that connection. So in terms of modulating the neuron connections, the way that it's adjusting that connection is by giving those neurons haircuts, sort of eating the extra connections that the neuron says it doesn't need. Complement system, yes, exactly. That is what I was thinking of, the complement proteins that assemble to make this attack complex. Thank you so much, Mac. Yes, thank you, A. It's kind of nuts. It really is like an assemblage of all of those immune proteins come together to form this weapon, much like a Gundam. It's nuts that's how that works. Complement system membrane attack complex. Thank you so much. Someone needs to label something biological a Gundam now. There might be something out there. I feel like... Let me Google that real quick. I feel like that might be something that already exists. What are the chances that I get something? Google Scholar might be the way to go. Gundam biology. Let's see what we get. Maybe not. It sounds like something that might have been a thing, or could be a thing. Yeah, you out there, you burgeoning biologists, you could be the one to make that a real thing, which would be very neat. Welcome back, Resta. I don't know if you're saying anything or if you've muted or unmuted a mic. Yep. Okay, there you go. We're back. Very nice.
Yep. Also, Borges, you might have some luck with naming novel phages.
Oh, to call a phage a Gundam? That could be kind of cool. Yeah, sometimes genes do have silly names. I did a whole YouTube short about running through some of my favorites. There's a story, and I don't know how accurate this story is because I've said this over and over again for years. It was a story that a professor told me in graduate school where there was a scientist who named a gene one-eyed pinhead in, I believe, the zebrafish. And one-eyed pinhead is a gene that produces anatomical disruptions in the developing zebrafish embryos. So I think it changed like the shape of their heads or something. So she called it one-eyed pinhead because they didn't have eyes that split. But also she added the pinhead part to describe her ex-boyfriend. I love that story, but the name was inappropriate because one-eyed pinhead is a gene that we also have. So the reason why that we study zebrafish is that they're a good model for developmental biology. And you never want your doctor to say, oh, I'm so sorry, your child has a mutation in the one-eyed pinhead gene. So they renamed it OEP, just the initials. Because, you know, you're out here and it's the sort of wild, wild west of developmental biology. You're knocking down genes, you're finding out what they do. It's crazy. There's a lot of an explosion of information at that time period. So you have all sorts of like weird Drosophila and zebrafish people naming all sorts of weird things, and it ended up biting us. Oh, it ended up biting doctors in the ass many years later. I've heard it on a site where if you discover a bacteriophage, you get to name it. It's good because phages have really boring names. Like the one that we looked at for cholera, it was just CTX phage, cholera toxin phage. Boo! It's kind of boring.
Yeah, there's I didn't take the class. It would have been cool if I could, but I know that some institutions offer classes where you can just go out into across campus and collect samples and extract phages from that. And because phages just have so much diversity, and there isn't like major pressure to have a super serious name. It's through things like this that we have the Cheeto Bro phage and the Uncle Howie phage.
Uncle Howie phage. That's cool. I didn't know that. That's really interesting. Phage minus prime. Cheeto phage. That's so cool. I love that. That's very neat. Wonder what models exist for comprehending the pattern of knock-on activations of a homeobox type proteins. I don't really know what that question means at a precise level. So what does the pattern of knock-on activations mean? So knock-on implies genetic engineering. So I'm not entirely sure how that connects with activation of homeobox proteins. There's a SpongeBob mushroom. Yeah. I think it would have been really cool, I think, to get to name an organism. Although, Resta, would you say that if you wanted to name an organism, that the most fruitful of those ventures might be somewhere in the archaea domain? You did call them like dark matter, or are we like having trouble with finding new archaea?
Well, for starters, uh, I think the naming of archaea is uh, is held to be a bit more serious. Though there are some really cool ones, like the Asgard archaea, where they have ones
Yeah.
named after Thor and Loki and other Norse entities.
And Chinese gods too. Sun Kong is one of them too. I was like, whoa, dude, diversity and inclusion, let's go. Still have lots of undiscovered fungi. That's true. That's true. Oh, fungi are cool. I love biology for being like the center point of the lived human experience, and I'm waiting for the day to talk about fungi with a fungi VTuber. It's only a matter of time. Surely one day I'll find one. I guess like there's a descending pattern of activations that determine organism shape in early development. Are we talking about Hawk's gene? Homeobox genes, yeah, that's what you said. Descending pattern that determines organism shape. Yes. So let me reread your other comment to understand the context. Comprehend the pattern of homeobox. Okay, I understand. So these Hawk's genes, homeobox genes, these are the genes that regulate your body plan. They're master regulators of why you have arms where you have arms. I did a short using Machamp as an example. Machamp having many arms. And so, yeah, gosh, that was like one of my first videos ever. Yes, the gradients set up by genes like Hedgehog and Wingless. So here's a teaser. And Marlon, if you stick around with this channel long enough, the end point of this Molecular Biology 101 series is developmental biology. For me, developmental biology is the final boss of biology because you have to learn everything before. You have to learn all of biology before you can understand how an embryo forms. All of cell biology, biochemistry, even bioinformatics to a certain extent, just to get to the point where you can even start to comprehend an egg, a sperm, a zygote becoming a fetus. And so your question about the homeobox stuff, I don't want to give you a bad answer now, but I will say that my goal, my final boss for this project of 101 lectures is going to be how to go from zygote to fetus. And Mandrake is correct in saying there are... this is making me want to do the Dunsparce video. I have a script for understanding why there are multiple segments for Dunsparce, right? There's the single form where you have one body segment. And then you have the Dunsparces that can have 2 or 3 body segments. And there's interesting genetic variation between those 3 forms. And that can be explained by homeobox proteins and gradients and stuff like that. So, but I want to give you an answer here. So say in a very simplified made up organism, you have an embryo that was fertilized and the sperm entered on the north side of the egg, we'll say. And we'll say in this made up organism that the sperm brings with it genes that activate your homeobox proteins. And so the concentration of those genes or those gene activators are going to be the highest where the sperm entered. This is a concept of cell polarity, establishing where the head is and where the tail is. So in this organism, you have a gradient where there's going to be the most concentrated signal for a certain program at the entry point of the sperm. And then as the molecules drift out due to Brownian motion, that signal starts decreasing in concentration. Right? And so one way that an organism can be wired up is, okay, the sperm enters the egg on one side and there's a high concentration of signal A. Let's say that at this threshold, just call it arbitrarily 100 power A, that in that spot, you turn on all of the genes you need to make a head. And at 50% power, you turn on the genes that you need to make arms. And at 30% power, that A signal makes legs. Of course, that's way oversimplified, but that hopefully gives you a gist of how that works, right? It's gradients and patterns and polarity. And it's one of my favorite things to learn, but my least favorite thing to get tested on because it is so complicated. I remember a pattern of this being responsible for the formation of endoderm, mesoderm, and ectoderm. Exactly. So those are the 3 layers that happen at a point in development where you start separating into cells with distinct lineages, cells that have had their cell fate written out for them, we'll say. This may be too much of a generic question, but what philosophy on life pushed either one of you towards the STEM section of academia versus the creative wing of life? Oh, Mute, I love this question. I don't know, Resta, if you haven't answered this immediately, so I'll tackle this first. I love this question because I was always torn between creative stuff and academic stuff, and For me, it's like rude almost to say that it was a money thing, but it started off as a money thing because I was so bad at everything that I wanted to do creatively. I never published anything that I did except fan fiction that I wrote when I was, 10. But I never got good at drawing. I never got good at any of that. And what I did get good at was studying. So at the beginning, it really was like affinity for a certain thing. And like, should you chase your dreams? Of course, you should to a certain degree. The more realistic way to pursue your dreams is to come up with a dream that you can do. And this is not my original dream. My original dream was to be Neil deGrasse Tyson, touring the country, lecturing, because I love writing scripts and talks, and I want to be a bridge between science and spirituality, not in a religious sense, but in a way for people to feel deeply connected to the study of life. That's what I wanted to do as a Neil deGrasse Tyson clone. But where life has pushed me, and this is a hobby I think that I will have forever, because it's not being Neil deGrasse Tyson, it's being me. I would never be able to do what Neil deGrasse Tyson does. I don't speak well enough in front of a live crowd to do what he does. And so, like, continuing to try, I've kind of fallen in to doing biology as a professional job, to stick with that as my main moneymaker, but to combine that with creativity. And so it's more on the science side, but I love that I get to express my love for science in a creative way. So it's kind of cheating because it's not neither nor, because I do have a foot in both worlds. But I want people so badly to understand that you can do both. So I don't know, Resta, do you have an answer for that?
Oh boy. This question. Well, my philosophy on life has significantly changed a lot, given my life experiences. Uh, there used to be a point in my life where, without a doubt, if you asked that version of me, I'd be like, yeah, I could totally be one of those PIs that lives and breathes and science is their like 99.99% of their life kind of thing. Because I loved learning. I loved experimenting, learning about new things. And like even at the time, I'd also have a bit of a creative side too, in the sense that I remember the first lab I worked in. We were taught how to do cell counts on an epifluorescence microscope, and like a week or two into the program, I suggested to my mentor and the PI, like, oh, if you try this, it can it can be a different way of counting cells that might be more optimal and also leaves a record of cell counts for future reference. Um, like, I guess that's also why I like bioinformatics as well, that engineering side of tweaking things to make it better. Um, and I'm definitely like that as well when it comes to OBS settings, my 3D VTuber model, uh, adjusting like all sorts of content creator things. And so I've had that curiosity and I've had that creativity for my whole life. Um, and again, for most of my life, I thought I was going to go like super full on hardcore academic, but without revealing too much, I'll say that there's been a couple life events that really have had me reevaluate some things. Um, and the importance I place on people in my life. And because of that, um, I'm currently in a bit of a career limbo where I'm trying to see if I can make content creation work as a full-time thing. And I'm trying out different strategies, but I'll give myself like a year or so for that to see how that goes. And if it doesn't work out, back to the lab.
I think there's an interesting wording in that question, Mute, in the sense where I don't really think that it's up to us whether or not our philosophy pushed us there. It's kind of like life just kind of happened, right? Like for me, it was the money, but that's not because of philosophy. That's just because, like, well, I didn't see a potential future where I could do creative stuff as a full-time thing. Like life kind of pushed me towards a direction. I think what you're explaining is also, shouldn't ignore the fact that, I don't want to paint a rosy picture of academia. I want to paint a rosy picture of learning, because learning is great and fun. But actually doing stuff your mileage may vary, right? Of course, your mileage may vary. So I think, getting pushed in is one way of looking at it, which might seem kind of horrible, but I think that, like, having life be done to you and take you in places you never thought you would go to can be even more rewarding than your initial plan. Because, like Resta, my initial plan was I thought I was going to live in science forever. And I kind of don't anymore. You could say that I'm still a part of academia, but I teach for a living. I don't do research anymore, right? I tried that life out, and life pushed me in a direction where it wasn't even an option to continue. I didn't really have a choice, honestly. For those of you who don't know, COVID shut down my research for 2 years during my PhD, so I never got to finish. So this idea, it doesn't look great for jobs if you spend 6 years pursuing a PhD and then you don't finish, right? Then they start asking questions. Of course, they understand because it was COVID, but because life kind of screwed me out of a normal biotech-ish job, I was able to land in a position where this is the best job in the world to me. And having the time to be here with you all is a feature of that job. I'll say, and I'll speak very personally about this, are there days where I regret or feel bad about not having finished a PhD? Yes. I think it's irresponsible to claim that I'm this enlightened being that never goes, damn, I wish I had a PhD. Wouldn't that degree look good on my wall? But at the same time, I have full benefits, decent salary, vacation, spring break, summer break, a YouTube channel that makes grocery money on the side, a lovely audience that hears me yap, and a space where I can talk about biology freely. Because here's the thing, here's the opportunity that this side channel gives me that my real job doesn't. And don't get me wrong, I love my real job, but at my real job there are parents who pay for their students to get the best education that they can get. And every parent has a different idea of what that means for their kid. And it is up to us as people that take their money in exchange for services to best match their expectations, we'll say. And that kind of environment can affect how I would want to cover certain kinds of content. Right, where here, just the fact that you've shown up into my stream, and if you've heard me yap for a while, I have been blessed with the opportunity to express myself in a very genuine kind of way, where I don't have to think about, oh, is this the best way to stick this in their head? In reality, what I want to do is this the most fun way, or the most fun and accurate and useful way to engage an audience? And to me, that is it is my artistry. I think I might not be able to draw or write fiction or photograph to save my life, as Toxo can attest to, but the opportunity to use my words to be an advocate for biology, it's nice. life is happening to you right now, at least in terms of work. Yeah, I can imagine it. It's happening for a lot of people, especially in like the tech realm, where life is just happening to you. And this isn't like a guru channel, so I don't have any advice to give, but just some sympathy that like, and hope that maybe getting pushed around by life isn't, mileage may vary, of course, but maybe it'll end up to be in a good place. I certainly hope that it does. Yeah, yeah. Did I end up getting a master's? Yeah, I got a master's. So the way that a PhD program generally works in my field, in the US at least, is you take your big boy exam sometime in your second year, where you get up and you get roasted by your professors, and they test whether or not you have the brain worthy enough to come up with a new idea and to come up with a reasonable plan. As long as you can do that and pass your classes, you can get a master's. Imagine if those parents found out that their kid was being taught by a neutrophil. I don't. This is why I want to keep my anonymity as much as possible, right? Because like I'm very confident in my material on this channel. I think what I do is great. As my regular chatters knows, it might turn some people off, but I'm gonna be out with it. I am proud of the work that I do. I think that it is good. Can it improve? Always, of course. I am not ashamed of my content in any way. Sometimes I just don't think that I don't want to say too much. People can disagree. And so I would prefer that they not find out what I do online. Do I have plans to cover stuff like cluster of differentiation membrane proteins? Don't know what those are. Maybe one day. Don't know what those are. And here regenerates over 8 days, technically replaced by a relative coming from the same stem cell blood lineage. Resta, you have a question in chat. Did you go with school in bioinformatics in mind? That's interesting. Yeah. Did you kind of fall into bioinformatics, or did you go knowing that you wanted to do that?
Good question. Well, when I first got into university, I originally wanted to be a marine biologist, living along the Pacific coast, just being exposed to the ocean and the life and craziness that it holds. But eventually, I I kind of fell in love with the whole molecular bio side, and wanted to know more about all the like super tiny molecular machines, and that really pushed me towards marine microbiology, which uh, I remember having a talk with one of my counselors. They're like, you're all over the place. You're what we call ghosting through majors, because I was taking classes from uh, like coding in Java and bioinformatic algorithms, and to marine geology and biogeochemical cycles, to cell biology and uh, um, like the more traditional biology route. So I had all these different kinds of classes that didn't fit into any particular major, but I knew I really liked microbiology. And so I pulled from all these different disciplines. And that's also something I like too, just how interdisciplinary both the marine sciences and bioinformatics can be. 'Cause I also took like oceanography classes too, of like studying the waves, studying the structure of a beach, studying uh, like global currents, and all of that. So it was I I mean, when it comes to learning, I'm kind of like a kid in a candy store. I just love learning and discovering new things. It's why uh, when I became a content creator, I'm just like, ooh, voice acting tips, ooh, 3D modeling tips, ooh, uh, OBS tips, um, just learning as much as possible. So I'm very much a jack of all trades in that sense.
I think that's an interesting conversation to have, especially the contrast between you and I, because, despite running this channel as an education channel, my main motivation to learn on my own is actually to make content. And it seems kind of soulless, but like, to me, the pleasurable part is to take very complicated information and package it in a way that is accessible and interesting, because it allows me to engage like the creative part of my brain. So like I did not want to learn how OBS worked at all. I didn't want to learn a whole bunch of stuff, actually. I'm a little more stubborn in that regard, but I love learning if I think that there's an interesting story behind it that I can share. So it's kind of an interesting contrast. Also, an interesting contrast of whether or not you should learn deeply or whether or not you should learn broadly, and I don't think there's one answer to that, but I think the meta right now amongst students is to learn deeply and not broadly, and that might be due to like economic insecurity, right? Like if you are looking for a job, and I really sympathize with the students who have to go through this, in times of economic hardship, of like, I want to be the most qualified student to get an internship, so I will do exactly what this company wants me to do and have the perfect resume to apply to that company. But like, and I'm not a career counselor, so I don't know the veracity of this situation, but I do know just from anecdotes, right, that you don't have to go super deep. You can take the approach of going broad and making the case for yourself that you're still the most qualified, as Resta has done. Like if you kind of octopus your way through and pick up everything, that can be good, at least in an academic setting. I don't know about companies, but in academia, if you have a broad skill set, I find that that's actually more beneficial than a narrow, deep, broad skill set, just because like, you don't know what information you need to know when you tackle research, right? And so going really deep into one field, that's useful. But when you're an undergrad, I really highly recommend that you take a couple classes that broaden your scope, because you won't have access to those later on. Then you're going to be a tool. You're going to be a tool. You're going to be specialized as a tool, as a one-trick pony, I should say. But if you have very broad, diverse interests, then you can always go deep later. I think it's easier to go broad in undergrad when you have this structure where you can learn a whole bunch of stuff. And then when you get into a research position or like a position where you are innovating, you will pick up the depth as you go along. I didn't know anything about neuroscience when I started my PhD, and now I like know a decent amount. I go as deep as I need to do the research that I needed, but because of my background in cell biology, that aspect of neurobiology has become a lot easier because I didn't choose initially to go super, super deep into one field. Yeah, just being able to have different ways of thinking is also great in terms of diversifying the different angles of approach you can have over a problem. I like what Morju says, that you almost willed your bioinformatics education into existence. I think that's very powerful.
Yeah. I feel like that's a common thread for lots of people in bioinformatics, because there's a lot of biologists who really don't like coding, and there's a lot of computer scientists who don't really care for biology. But me being kind of like the octopus that I am, found that the connections between the two were really cool. In fact, this reminds me of one of the coolest revelations I had when I learned it in school, which was how Le Chatelier's principle of equilibrium that I learned in chemistry relates to the phosphorylation and dephosphorylation of ADP and ATP, and how life uses that reaction to make non-spontaneous reactions that are necessary for life actually occur.
I think that's poignant in a way where I had a similar conversation with Clockwork over like when you first learn photosynthesis, there's a bit of extra insight that I think teachers should really include in their teaching of photosynthesis, because depending on where you go, you just learn what photosynthesis is. It takes sunlight and these ingredients, and you end up with sugar, and that's cool. But as you said, with like understanding the fundamentals of like, this is the reaction that drives what life actually is, and wording photosynthesis, like, you have this fundamental energy problem, that energy can neither be created nor destroyed, but the forms can be changed. And when you spend a unit of energy, that energy is lost to the environment as heat. So it's kind of an interesting chemical idea that as life continues, you are slowly degrading. I'm sorry that you have to hear it from me like that, but you are slowly expending energy. The entropy in your body is increasing, your telomeres are shortening, your life is shortening, you're turning organic matter and using it and burning it for fuel. And so if that system were to be closed, then we would all eventually burn out. And the thing that makes it so that we are not a closed system on planet Earth is the sun. Because again, energy can neither be created nor destroyed. And so if we're constantly using energy, there has to be a source for that energy, and that is literally the sun. Even though we think of plants as being sun powered, but all of the biological energy that you have access to is only there, or for the most part only there, because of the sun. So it's more like we are also sun powered, because if the Earth were a closed system, we would all just degrade into nothingness. There's a lot of poignancy. I think if you're a romantic in science, a lot of things that you can find great and deep meaning. And there's the existential dread. Let me tell you, when I first learned about telomeres in genetics, that was my oh crap moment. Like, you're saying that, like, I don't know the number, Resta, but there's a significant percentage of your DNA that just jumps, whether or not you want it to. And to me, that really, I couldn't like sleep for a while. That's so freaky. Yeah, transposons, exactly. That's what we're saying, transposons. Yeah, in terms of a little bit of an insight, in terms of, and I think Marlon has been suggesting some good, interesting ideas for future streams. The way that I choose what to cover is actually not based on the content, but whether or not it forces the audience to think in an interesting way. I think there are plenty of other channels that will just give you interesting things in biology because the facts themselves are interesting. But I hope if you're a longtime viewer here, then you're staying for the fact that, Resta's asked some really interesting questions to chat today, and we've had some really interesting answers, and answers that go beyond the scope of what we're hunting for. And that activity of forcing yourself to say new things is valuable. And so I'm not saying that I won't cover those things. I'm going to say that depending on how complex the papers are, it might not make for the best journal club streams, because I like to do the thinky bit. The thinky bit is the interesting part. Believe it or not, software engineers have those moments too. Wait, seriously, this library that's held together by literal duct tape is the most important thing that makes our company money? CrowdStrike. Who's up for CrowdStrike? Yeah. That was a fun thing to wake up to. Pour one out for our IT bros. Pour one out. Okay. Okay, so my voice is starting to get a little hoarse. I just wanted to plug one more time, and then of course thank Resta. Next week, we are going to be having my 1-year anniversary. I'm going to go ahead and show my PowerPoint again. And hide your PowerPoint. Yep. So that I can talk a little bit about how that's going to work. All right, presenter view. Yep. Yep. So we've made content for a whole year, which is very, very exciting. I have a whole lot of fun things planned for this 1-year anniversary, including things like looking at data. We love data in this house, as well as looking back fondly on some like milestones. We blew through, I think, where I thought I would be at the end of a year, it's just been an incredible journey so far. And sort of looking back on those times, I think is going to be really fun. But also, For members, we are going to look at the actual first piece of content that I made on the Phi the Neutrophil channel before it was even called Phi the Neutrophil. We are going to look at a video I did on a probiotic that claimed to give you larger testicles that I covered in my video about kombucha. So for those of you who don't know, I will never make any educational content behind a paywall. I think that is antithetical to the goal of an educator, and this isn't my full-time job, so I don't have to. So if you've watched that kombucha video, you already know that I talk briefly about this probiotic that claims to give you bigger balls. I just said it, whatever. That's actually the first piece of content I made before I was Phi the Neutrophil. And so after this 1 year anniversary stream, we're going to do a members only stream where we kind of live react, or I live react to watching that video. It is very different from my current voice, which I think will be interesting to hear. I've also gotten comments from people that I've sent it to, because it's unlisted on my channel. You can't watch it anywhere. I've gotten comments from clips of it, from clips of that era of my life where people have said that I've sounded a lot younger back then. Maybe that's true. But also, getting a peek into an alternate universe of what Phi the Neutrophil could have been, I think will be very fun. But also during this stream, I'm also going to talk through milestones. Maybe Toxo will show up and we'll like chat. I'm going to have a slice of cake to celebrate and some tea or boba or something. I don't really know yet. And then something that's very exciting for me personally is I've invited some VTubers to contribute little birthday gifts. And so I want to like open gifts live on stream and talk a little bit about or reminiscing about the collaborations and the content that we've made over this first year of the channel. I don't know how many I'll get. I think it would be very funny if I didn't get any. So like I've mentioned to all of the VTubers that I've invited, you don't have to give me a gift. I'm not going to be sad or upset. I think it would be hilarious if I didn't get anything. So there's that too, as like, you know, content pilled that I am. So just to set the expectation, I don't know that I'll open gifts, but we'll all find out together whether or not I have a very lonely birthday party. I hope I don't, but I also wouldn't be mad if I did. So that's going to be really, really fun. By the lonely Phil, exactly. Doppel is stored in the balls. That's true. Doppel is stored in the balls. Very, very good. So with that in mind, oh, you've seen the ads. Oh, Mandrake, you've actually seen the ads. Yes. So for those of you who don't know, I don't know if you know, but I actually got these ads as a graduate student. It's this guy who paid for these ads that were aggressively like YouTube ads too. This was an ad on YouTube, which is what inspired me to make that video. This idea that they're selling this probiotic that can augment your manhood in certain ways. So it's really fun to see that someone else has seen that advertisement. It's absolutely crazy. Okay. So yeah, so that's happening next week. Resta, do you have anything to plug before I hurl compliments at you and thank you at this very end of the stream?
At some point, I know I'm gonna have a lecture-based stream about introduction to deep sea microbiology. That's gonna be interesting. No specific date on that yet, but I'm hoping to push it out sometime this year.
Yeah, we can make sure that we plug that, and of course you'll let me know when that happens so that I can blast it all over my socials, because you already know, I did an Archaea video. It's something that I am very interested in, so we'll make sure that you hear about when that happens, and one more time, thank you, Resta, for taking the time out of your busy schedule to put together this very well-made presentation. I think you did a fantastic job. I really look forward to your microbiology video or stream in the future. I think you did a great job. I hope you had fun on the channel. I hope chat has had fun. I could not have done this bioinformatics stream without you, so I'm really happy that you've allowed me to throw a bone to all the computer nerds in my community, because for some reason I think I do have an overrepresentation of computer people in my chat. So I was really meaning to do something for them, but I couldn't think of what to do, and you've really allowed me, I think, to give them content that they could more relate to than the stuff that I've been pushing out. Yeah, that was awesome, Resta. Thank you so much. With that being
Thank you for providing this opportunity for me too.
yeah, and I don't know if you can hear, but Brakinji, my cat, is also celebrating. Okay,
And
bye,
thank you too,
y'all.
Chad.
Yeah, thank you, Chad. Thank you, Chad, for showing up.
Bye-bye.
Bye.
Bye-bye.